So shall we start?
Yes, yes, you can. Please start.
Good evening for Indians and very good morning for Americans. It is an honor itself to honor the beautiful audience who has gathered here. I, Professor Dr. Archana Tiwari, Dean Faculty of Applied Sciences and Director School of Biotechnology and Autonomous University Teaching Department, Rajiv Gandhi Pradyogiki Vishwavidyalaya, University of Technology of Government of Madhya Pradesh, accredited with Grade A by NEC, Bhopal, welcomes you all on behalf of the whole Global Biotech and Madhya Pradesh Council of Science and Technology family on International Virtual Colloquium on Novel Insights in Molecular Medicine in the Unexpected Pandemic Period. Thank you all for giving us your prize time. Dean William once said, "The aim of education is the knowledge, not of fact but values." On the auspicious occasion, we get opportunities to test our knowledge and understanding. We look forward to getting exposure to what the best of the brains think about these very dynamic issues of molecular medicine. Before the launch, let me first acknowledge the presence of some of our eminent guests, which are the galaxies of intellectuals, our excellencies and dear participants. As a tribute to Ma Saraswati, the goddess of knowledge, Ma, we pray for your blessings with all humility. Give us the wisdom to make this world a better place. May all human beings live in harmony with nature and other forms of life. May the truth prevail. May the darkness of ignorance be swept away by the dawn of self-realization.
अर्चना तिवारी जी, आप अगेन you are muted. Just unmute your mic. Please unmute. Still muted. Archana ji, your mic is muted. We cannot hear you actually. Just please unmute your mic. Unmute your mic.
Now, am I audible?
Yes, yes. Now,
Yeah.
now you are.
I'm very sorry for that. Molecular medicine aims to reveal molecules such as genes, transcripts, proteins, and metabolites to underlie the mechanism behind the physiological process, as well as alterations during the pathological conditions at the cellular level. Furthermore, molecular medicine intends to improve public health care and disease management through the development of biomarker-based screening, diagnostic and monitoring systems, as well as the target and mechanism-based treatment. That is, treatment strategies. Human Genome Project has been completed in 2003, exactly 50 years after Watson and Crick invented DNA structure. Based on this valuable breakthrough, the 21st century's molecular medicine approaches have been attributed to identify and understand functions and interactions of human genes to shed further light on health and disease mechanisms at the basic molecular and cellular levels. Published by James Watson in the first edition of the Molecular Biology of the Gene in 1965, the central dogma of molecular biology was a complete demonstration of the flow of gentle information described as DNA makes RNA, which in turn makes proteins. However, later in the 1980s to 1990s, by applying improved molecular biology methods, the single gene and inheritance concept has changed to multiple genes and inheritance with interactions of genes, RNAs, proteins, and the environment in a particular cell. Extension of central dogma has led to the proper and critical understanding of diseases and the generation of molecular medicine. By this way, a new concept called phenome as the total phenotypic characteristics of an organism has emerged, which implies the interaction of the whole genome with the environment. Primary objectives of molecular medicine include predicting potential future pathologies, identifying disease state through effective screening and early diagnosis system. The decisions on effective treatment strategies, monitoring the prognosis and health care, and predicting recurrence earlier to apply alternative treatments. In this regard, molecular medicine aims to obtain decreased under or over misdiagnosis and generate effective targeted therapies without side effects. Here, we provide an overview of the latest updates of molecular medicine, including promising research strategies and their emerging roles in biomedical research in the unexpected pandemic period. The evening or morning is a wonderful blessing, either cloudy or sunny. It stands for hope, thoughts, and giving us another start, what we call life. Once again, a very warm welcome to all of you. Today is our guest of honor, Professor Dr. Anil Kothari. He is Director General of Madhya Pradesh Council of Science and Technology and Scientific Advisor to Government of Madhya Pradesh. Unfortunately, due to some unavoidable circumstances, he is not joining the meeting now. We would request the most honorable personality of today's evening, His Excellency, our chief guest of today's function, Honorable Vice Chancellor, Professor Dr. Sunil Kumarji, the sculptor of human character, a seasoned scholar, and a navigator of this flagship of knowledge, for his blessings. I would like to request our Honorable Vice Chancellor, sir, to shower your blessings. Honorable sir, over to you.
Dear friends, first I would like to welcome you all on behalf of RGPV in this international colloquium, and which is being organized by Department of School of Biotechnology of RGPV. As you all are aware that we are going through this very tough phase of life wherein we are struggling with this pandemic called COVID-19. Whenever you talk about something which is of fundamental nature, then you require some fundamental research also. As far as my understanding of virus is concerned, unless you go at the molecular level, it's too difficult to fight these kind of viruses. So definitely any research which is of fundamental nature and which is done at molecular level is definitely going to yield result. And it has happened earlier also. Whenever we have struggled in such periods, only then you start remembering those fundamental type of research wherein you feel like it that something else or something novel has to be done. So you cannot underestimate or you cannot say that the time of fundamental research has gone. Every time you are realized or every time you are remembered again and again, unless or otherwise you do the fundamental research, you may find something new which you will not be able to handle with the kind of technology which is available as of now. So this is a time again and again to appreciate this fact. You cannot underestimate this fundamental research. So again, I have just on behalf of RGPP welcome you all, and I hope the kind of discussion which will be going in this particular colloquium may result some fruitful insight on the fundamental research on molecular medicines, which may be helpful for ultimate elimination of this COVID-19 virus, or that may also prepare us to struggle it out and fight it out in a better way. Thank you very much for inviting me in this call. Thank you.
Thank you very much, sir, for your thought-provoking address. Set a perfect platform for us. Slowly and gradually, we are moving towards the session. First, that is, we are profusely elated to take an opportunity to introduce our first eminent speaker of the day, Dr. Sushil Ji Rane. He is the Section Chief of Diabetes, Endocrinology and Obesity Branch, National Institute of Diabetes and Digestive and Kidney Diseases, Health Information Center, National Institute of Health, USA, one of the #1 growing organizations of the current era, for his converse. He is our oldest and important collaborator and a very humble person as well. Besides, he is also known for his work in different fields such as health, education, and research. He is going to deliver his talk on bridging neurons and pancreatic islet beta cells. Over to you, Dr. Rane.
Okay, thank you. I'm going to start sharing. So first of all, thank you very much for the organizers and for giving me an opportunity to share some of the work. It's really interesting. It's an interesting time, as was mentioned before. It's a very, we're all trying to understand how to use this time more productively. So, so in a sense, everything that we're doing on a daily basis has been fruitful, because eventually all of these sort of changes that we're making to our lifestyle, as well as how we do science, how we think about science, and how we interact with scientists around the globe, has already changed. So in a sense, there's going to be some positives from all of this. So having said that, I'm going to take this opportunity to share with you some research that we have ongoing for the past 4 or 5 years, actually. And we're almost to a point where this paper is going to be in publication very soon. So I wanted to share with you some of the, it's a story. Although it's a long talk, I really would prefer it that it be a conversation. My purpose is to just give you an idea of what we are trying to do. Why did we start this project? What was the thinking behind it? And hopefully I'll be able to convince you that some of the data that we have is going to lead into some new knowledge about diabetes, about how glucose is regulated, how insulin is secreted, and things of that nature. So, so this slide, for example, the title slide, it basically is trying to merge two very different cells. So on your left is, I'm going to turn my laser on here. Okay, so for example, so this is the pancreatic hypothalamus, or rather the hypothalamus in the brain. So this is the hypothalamic region in the brain that's regulating our desire to eat or are desired for hunger or satiety. For example, it's 6:30 or 7 o'clock in the morning in the US. So at this point, I'm not as hungry, but what I'm going to do is eventually have breakfast at some point. On the other hand, in India right now, it's close to dinner. So our physiology, both in India and US, is completely different at this time. So in your case, you're preparing yourself for a big meal, which is the dinner. In my case, my body is preparing for a small meal, which is a breakfast. So in all the times, I just got up after about 5-6 hours of sleep. In your case, you've been working throughout the day, or you've been active throughout the day. So both our body's physiology is very different. And we need to understand how the physiology works for us to understand how our body works and how we can prevent disease, especially such as diabetes and obesity. So here is the brain and the hypothalamus, which regulates glucose levels in our body. On your right is what's called an islet. It's in the pancreas, and the islets mainly have beta cells that produce insulin. And insulin is the only hormone that can control glucose levels in the body. So this whole context of the brain deciding when to eat, how much to eat, when to stop eating, and the beta cell, which secretes insulin that will control glucose, has always been suggested that there is a connection between these two cells. Of course, the brain is at the top of your head and the pancreas are deep down near the stomach. So these two organs are completely separated from each other. However, it's known, or it's at least been thought of, that these two cells connect in some way and regulate your ability to eat food or to digest food or regulate levels of glucose in our body. So what we are trying to do is try and understand how this happens. So, and again, this is something that was done long time ago. So this is in the 1800s. Ivan Pavlov was a Russian scientist who went on to win the Nobel Prize long time ago. And he was interested in studying physiology, human physiology. And in his case, his model system were dogs. So he used to work on dogs to try and understand how the digestive system in dogs work. So here he has, in his lab doing work that what we do with mice or rats or whatever. So, and one of the main things he noticed was he used to have these dogs, in a cannula to understand how the pancreas secretes enzymes. So here you have a cartoon of this dog connected to a funnel, which is releasing enzymes from the pancreas. And this was a completely different type of work. But what he noticed is periodically when food was presented to these dogs, the dogs started to salivate, so just to produce saliva by when food was presented. The other thing he noticed, the dogs started to produce saliva even when people came into the room with the food. So what he thought was, these dogs are anticipating food. So what he tried to do is do a very clever experiment. What he tried to do was present the food and then ring a bell. And the dogs got used to it, saying that the bell is associated with the food, and the dogs started salivating. And then finally what he did is took the food away. The food was nowhere near the dogs. All he did was rung a bell. And as soon as the bell was rung, the dogs started salivating. So dogs were associating the bell ringing to the food coming in. So that was the first demonstration that our psychology is already designed to anticipate food. So when we go for a wedding function or any other function where there is a meal, we already are anticipating there is going to be good food. When you're celebrating Diwali or any other festival and you go to somebody's house, you're already anticipating there is going to be sweets. And this is a very interesting phenomenon because our brain is already designed to recognize when there is food. What type of food is it? So that this level of anticipation has been known for hundreds of years, but we've not been able to study it. So now with the tools that we have available, we can study something like that. So that is the main focus of this work. And again, going back on history, as was said before, history sets the foundation for research. So this is another eminent scientist from France, Claude Bernard, and he did a lot of important work. One of the ones was very basic, but it was very unexpected. So sometimes you don't know what you're going to find from unexpected experiments. So in this case, he was working on understanding the anatomy of the brain. And what he did was he ended up puncturing the fourth brain ventricle near the hypothalamus. By just putting a needle in the brain, he just punctured it. And what he saw was just that one elimination of that fourth ventricle led to an increase in glucose levels in his rats. So there was another connection where something done to the brain led to a change in glucose levels. So all of these evidences done over 100 years ago suggested that our brain does control the levels of glucose in the body. It's just we did not know until very recently how that's happening. So now I'm going to go a little bit into my talk. So for example, here is just a cartoon of a mouse. Just to orient you, this is the pancreas, and the pancreas has what I mentioned are islets. The islets have what are beta cells. The beta cells are the only cells that produce insulin. Of course, insulin is required to control glucose. So far, for the last 100 years, most of the research on insulin has been focused on beta cells. And naturally, it's a very important cell type to study. We also study that in my lab. What is also known, though, that these islets are also connected by neurons. So here you have a neuronal connection that's coming from the hypothalamus. So this is the brain region in the mouse, and this is the hypothalamus of the brain. And what you have is nerves that come from the brain and are coming close to the pancreas and coming almost close to the beta cells. So here you have evidence where neurotransmitters such as acetylcholine and noradrenaline are being released near the beta cell, and they have been known to control insulin secretion. So what is presumed, although not confirmed until now, is the beta cells have a bridge that connects them to regions of the brain. Now this makes more sense, because when we think about food, or when we think about eating, or we think about not eating, it is good to know that there is a connectivity that might happen from the brain to regions in the body that are involved in digestion, such as the pancreas. So it's always been known that there may be a connection between the beta cells and the brain, but there was no direct evidence for it. And now what we are trying to do is try and find out, is there a connection and what does that do? So here is the hypothalamic neuron, and here is the beta cells, very far apart, but seems like they're connected through these wires. It's like an electrical circuit in any television or a radio transmitter. It's no different than any circuit. So this is a neuronal circuit. And what people have done in the past is using very, technologically not the best techniques, such as lesions, such as damaging the brain, or adding chemicals near the brain, or giving electrical stimulation. They've shown that changes of any of these natures, electrical lesions, chemical or physical or electrical stimulation, changes levels of glucose. That's been known. But other than that, not much else. So what we are trying to do is understand how the anatomy of the pancreas talks to the brain. And the way we are doing it is using viruses. Viruses, just like was discussed with COVID-19, these are amazing viruses that have the ability to infect cells very quickly. And biomedical research uses these viruses as tools to do research. So in this particular case, although this project started 5 years ago, it's interesting that I'm now going to be talking about using viruses to help us understand biology. So these are retrograde viruses. What I mean by retrograde viruses is they will infect one cell and go upstream and travel to the next cell by just infecting the next cell that is connected to the prior cell. And that's how that works. So here you have, if you inject this retrograde virus in the pancreas, now you'll see that virus will just travel upstream to wherever the neuronal connection is, and eventually it will reach the brain. And then you're able to find out where the virus went. So the question we are asking is, which of the hypothalamic neurons, as you know, in the hypothalamus, there are a lot of neurons doing a lot of other functions, such as feeling, touching, smelling, seeing. All of this is a neuronal function. So the neurons are doing multiple things. But what we want to know is, which are the neurons that are now connected specifically to the beta cells, okay? So, and the second thing is, once we are able to find these neurons, can we change these neurons? Can we manipulate these neurons? Can we activate these neurons? Can we inhibit these neurons? And then ask what happens to the beta cells. And the third thing is, you change the level of glucose in the body, such as by eating or by starving. Change the level of glucose in the body, and then ask if these neurons, the activity of these neurons will change. So there are three very simple questions, of course, questions that took us 5 years to get answers to. So the first question is, identify the neuronal populations that communicate with beta cells. The second question is, investigate the role of these neurons in beta cell function. What I mean by beta cell function is insulin secretion. And the third is, study if these neurons respond to changes in glycemic conditions, such as extra feeding or fasting. And how we are doing this, as I mentioned, is by using these viruses, which are fluorescent labeled. So you have GFP attached to these viruses. And so you can see where the virus is going from the beta cell. Where does it go? You can section the brain and look for GFP signal in the brain or anywhere else. The second thing is, once we find these neurons, now there is technological superiority in the sense that you can identify those neurons and selectively activate it or selectively inhibit it. And then you can see what happens to the beta cell function. And the third is, you change the level of the glucose in the body and measure the activity of those neurons to see if the neurons sense glucose. So going to the first one, so what we have is two types of retrograde viruses. One which is very limiting in a sense that once you infect one cell, it goes to the next cell and it stops. That's it, it's done, its job's done. It cannot go anymore. What we need is more than that. What we need is called as multisynaptic viruses that can travel from one synapse to another, because nerves are connected to each other through synaptic junctions. And so what we have is a virus that goes from this neuron, it's infected in this neuron, goes and infects the next neuron, and then goes and travels to the next neuron. And that's how it will proceed. So what we are using is this type of multisynaptic tracing to figure out how the beta cells are connected to the neurons. So this is just the structure of the virus. I'm not going to go into details with this, but what we have done is, since we are interested in insulin production, we are only going to be interested in beta cells. So for that, what we need is a mouse that only will express an enzyme in the beta cells. And so this is a mouse that expresses insulin and has this enzyme pre-recombinase that's going to be expressed only in the beta cells. So what we are going to do is inject this virus in the pancreas, like I'm showing here. Okay, so it's going to infect all the pancreas. So the virus infects and spreads rapidly. Once you inject it, it's going to infect the entire pancreas. And then what it's going to do is go to the beta cells. So it will infect the beta cells, you can see the beta cells becoming green here. So this is a immunofluorescence using confocal microscopy, showing that this virus has now infected the beta cells. And then what's going to happen is it's going to go out of the beta cells. Because this is a virus, you cannot stop it. It's going to keep going, and eventually it's going to reach the brain. And once it reaches the brain, after a couple of days, then you go and study what happened. All right, so this is the neuron. Now this virus has infected this neuronal cell in the brain, as can be seen here. So this is the primary infection of the eyelid with GFP signal. And this is the ultimate infection, which is in the neurons. So you can see the neuronal cell here with these projections, the synapses also showing a green fluorescence. And that's because the virus has arrived here after 2 days. So here is how we go about it. We look at all these sections from pancreas to see where the virus has traveled at different times. This is 72 hours. So this is an eyelid stained with insulin. This is with the virus, showing that the virus is definitely going into this beta cells. So here is your pancreas staining. Different regions of the pancreas are staining this virus. But most importantly, what we want to know is what is going on in the brain. So this is the brain of a mouse, and then you section that. So you section using your cryotome, different regions of the brain, and you'll see in all the regions of the brain, after 6 days, you see this virus appearing. So all the green signal is only there because the virus that was injected in the pancreas is now traveled to the brains and infected the brains. So this is how you know that the beta cell is connected to the regions of the brain. Then we go on and ask which regions of the brain are important. I'm going to skip over a lot of the data here. What we find is there are two main regions in the brain that seem to connect to the beta cells. One of the region is called the paraventricular nucleus of the hypothalamus. So it's down deep in the brain near the hypothalamic region. This region is very important because it is involved in appetite as well as satiety. So our ability to eat a lot of food or our ability to stop eating, it's all regulated by this region in the brain, which is in the hypothalamus, called the paraventricular nucleus. So what we find is this green signal is appearing in the hypothalamus very strongly after 6 days, suggesting that this may be the region where there is a connection. Of course, as I said, there are other regions as well. So to cut a long story short, what we ended up doing was identifying that there are neurons within that region that secrete this peptide oxytocin that are involved in regulating the beta cell function. What we find here is these viruses are co-localizing with this hormone oxytocin, suggesting that some of these neurons are positive to this oxytocin, oxytocin-containing neuropeptides. Now, so what we did is find that the beta cells are physically connected to the neurons. And now we have identified these neurons. Now, what are you going to do with it? So the next thing we want to do is take those neurons and activate them or stimulate them, or you can inhibit them, silence them, and then ask, does the beta cell function or insulin secretion change? So this again is something that we would do using viruses. So what we now do is we are not injecting the pancreas anymore. So there is no injection in the pancreas now. Now we take this mouse, which will have only oxytocin neurons, and we inject a virus here in the brain. So this is using fiber in the brain, cannula. And what we do is inject this in the brain. This virus will only get activated once we inject this intraperitoneal something called as clozapine N oxide, which is an agonist. So once that's injected using IP dose, it will travel to the brain and will activate these neurons, only these neurons in this region. As you can see here using signal here with the red fluorescence, we know that this virus is expressed. So once you activate this, now you can study this mouse, what happens after you activate these neurons. And the way we do that is fast the mice for 16 hours. So 16 hours, this mouse had no food. At time 0, we measure the plasma, measure the level of insulin. And then at time 0, we give it glucose. Glucose alone or glucose with this drug CNO, which will activate these neurons. And after 10 minutes, you measure the plasma, look for insulin. And what we find is after 10 minutes, there is activation of neurons and there is inhibition of insulin. So if you see from the graph here, compared to the first 0 minutes, after 10 minutes, there is a reduction in insulin. So the bottom line of this experiment is when you activate these neurons, less insulin comes out. So insulin is suppressed. So inhibition of the activation of these neurons reduces insulin, okay? And if you reduce insulin, your glucose should go up, as we know. When insulin is low, glucose goes high. So your next experiment is measure levels of glucose. So for that, what we did is implanted in the mice a device that will measure glucose. And what we did again is remove the food from the mouse right here. So it's fasting. And at 0 minutes, again, we give glucose or glucose with the drug that will activate the neurons and then measure the glucose levels minute to minute, measure it. And so every minute you get how much glucose is produced in that animal. And what we see here is once the neuron is activated, the glucose levels go up. It's shown here in this graph, and it's also shown here in the bottom here in this particular graph as well, suggesting that as soon as these neurons are activated, we already saw that insulin levels went down. Now we see that the glucose levels went up. Of course, that's what happens when insulin is down, glucose is always high. So this is your confirmation that activation of these neurons is going to increase glucose levels. We did another experiment where the mice were fed for 4 hours, and then you give it food and then measure for about 3 hours their glucose levels. Again, when the neuron is activated, the glucose levels are really high compared to the control, your saline injection. All of this suggesting that when you activate these neurons, insulin is reduced, glucose is increased. And that's all happening by activating neurons in the brain. And then second experiment we tried is what happens when you inhibit them. As I mentioned, these neurons release oxytocin. So what we want to do now is to inhibit this neuron. So what you do is what happens when you just stop this neuron from activity. So again, we inject a virus in the brain that will inhibit these neurons, and same experiment. Now we're going to again inhibit these neurons and look for insulin and glucose levels. So here is the inhibition experiment. So once you inhibit these neurons, now insulin levels go up right here. So here you have control, here you have using the virus, there is insulin levels go up. And then when you look at glucose, opposite to insulin, glucose is going down compared to the control. So the neuron activation increases glucose, neuron inhibition reduces glucose. It's something by just changing the activity of one set of neurons in the brain. So this is the summary for that. When you lower insulin, and when you inhibit these neurons, you increase over here the insulin. The third and the last experiment is measure the activity. So what we want to know is, can changing the level of glucose in the body change the activity of these neurons? So here what we designed is hypoglycemia. When do you have to suppress insulin? You suppress insulin when you're fasted. So when you're fasting for a long time, if there is a puja and you're fasting for an entire day, now your body has depleted glucose. At that time, because your glucose level is low, you have to keep insulin down. If at that point insulin is high, your glucose is going to be even further, you're going to become hypoglycemic. So what we designed is look for conditions when there is low glucose. And the way you can do low glucose is using this compound called 2-deoxyglucose. It's a metabolite of glucose, which induces hypoglycemia. It looks like glucose, but it doesn't function like glucose. So what we do is inject this 2-deoxyglucose in the mouse intraperitoneally, and then section the brain and look for activity using a transcriptional factor for CFOS. CFOS is a marker for neuronal activation. And what we look is how many neurons are activated in response to this 2-deoxyglucose, which induces hypoglycemia. And what we see is once you inject 2-deoxyglucose in the body, the mouse becomes hypoglycemic, which means there is very low glucose in the body. These neurons are activated much higher compared to the control saline. And the reason why these neurons are activated is that so they can suppress insulin. Because when the glucose is low in the body, you do not want more insulin coming out. That will create hypoglycemia and comas and unconsciousness. So this experiment shows that when you have low glucose, the neurons are activated much more compared to the control. We similarly did this exact experiment using another very novel approach where you can image the brain. So our neurons, when they fire, just like electricity, when neurons activate, there are changes in calcium signals. And you can measure using a camera on the brain these calcium signals and detect activity of the neurons. So what we did here, again, is image the neurons activity using optical fiber. And what we do here is, again, inject the 2-deoxyglucose, which will induce hypoglycemia. Hypoglycemia will shut down insulin, which means that neuron has to be activated. And so we monitor minute to minute the activity of these neurons. And what you see is once this 2-deoxyglucose is given, either 200 milligrams or 400 milligrams, there is increased activity of these neurons compared to the controls right here. So this is 200, this is 400. What you see is the neuronal activity is increased when the mouse senses that it is hypoglycemic. So this was the final slide. When you activate, when you have decrease in glucose, these neurons get activated. So just to summarize my talk, so what we have done is we have identified something that was proposed hundreds of years ago, that there is a connection between neurons in the brain and beta cells that produce insulin, and insulin regulates glucose. What we have done is then investigate the role of these neurons in beta cells and study if those neurons respond to changes in glycemia. So what we find is there are indeed hypothalamic neurons that are connected to the beta cells, and these are releasing oxytocin. This is just some of these neurons, not all of them. I'm sure there are more, and we are going to keep working on it to find more neurons that are involved in appetite and glucose regulation. And what we find is when you stimulate these neurons, you suppress insulin. When you silence or inhibit these neurons, you increase insulin and you lower glucose levels. And then when you give a hypoglycemia signal using 2-deoxyglucose, what we find, these neurons are activated, suggesting that they are protective. So when your body has very low glucose, our body has designed a system that when you are extreme fasting, your body will talk, your stomach will talk to the brain and tell it to shut down insulin, because you do not want more insulin to come out already when your glucose levels are low. So I'm just going to finish by acknowledging some of the people who did the work. Ioannis Papazoglou is a visiting fellow from Greece who's done most of the work. And of course, there are other collaborators that we use for some of these things. So thank you again, and I'm happy to take any questions or discussions.
Now the talk is open for the discussion. If anybody is having any queries, please ask. Free to ask.
So just, can I ask a question, listening,
Sure.
Lalit?
Sure, Dr. Lalit, please.
So how far the daily activity, like when you have a meal, you have like insulin secretion and glucose goes in and out. So does the meals also affect the hypothalamic neurons, or is it insulin directly act on them? How do feedback regulation?
Yeah, so that's a very interesting question. Exactly. So I think that is where this work will go. So as I mentioned, so right now, both for you and for me, it's around 8 o'clock in the morning, and our body right now is very low in glucose because you've been sleeping through the night. So the body has to recognize this, that you cannot have more insulin in your body right now because you already are low in glucose. If you keep putting out more insulin, that low glucose is going to go even further down, and you're going to get fainting, unconsciousness, blurriness, a lot of the things that happen when your glucose levels go down. At the same time, when you eat lunch, dinner, you go to a buffet, you go to a wedding reception, and you eat a lot, at that time, the body has to recognize that I need to put more insulin in the system because I'm eating a lot. I'm eating a lot of mithai, I'm eating a lot of, whatever. Yeah, last night I had chole and puri, means I know my insulin levels were very high. So our body has to have what is called as a safety valve. Like when you have a pressure cooker, there is a safety valve. So otherwise the system is going to blow up. So the beta cells in the pancreas have a very delicate job. The delicate job is to produce insulin depending on how much glucose is in the body. But you don't tell the beta cell when you're going to eat. You don't tell the beta cell how you're going to eat, how much are you going to eat. So somebody has to prepare the beta cell for that. And I think evolution has prepared that there is a safety valve, which is in the brain, which will sense, which will feel, which can touch. You smell food. The moment you smell food, you go to a buffet, you will start salivating. You know what's coming. The food is coming. So our brain is now wired to these organs that eat food, that digest food, so that we can protect from ourselves from either overeating or we can protect from not eating for too long. Both are very, very important. It's very amazing that glucose levels in the body are maintained between 70 and 110 milligrams per deciliter. If you go below 70, you're going to get unconscious. If you go above 110, 120, you're going to be diabetic. So very narrow range. So insulin production has to be very tightly controlled. And what we think is what has been proposed is all your daily activities, your exercise, your eating, your fasting, everything is going to be talking to the brain so that the brain can control how much energy you have in your body.
Sakshi Bharadwaj has raised some queries that is in the chat box. Dr. Sushil, kindly go through the chat box, please.
Okay. Now, can you read? Is it possible for you to read that? Because
Yes, sir.
Will the diabetic condition affect the activity of microflora of the digestive tract? This is the question from Sakshi Bharadwaj.
So this is, you mean the neuronal activity?
Yeah. Will the diabetic condition affect the activity of microflora of digestive tract?
Yes, I think that is definitely the microflora regulation is again a completely different field. In fact, Dr. Yadav is going to talk about that much later today. But yes, the diabetic conditions are known to change the gut microbiota in the intestine, and that itself changes a lot of hormonal regulation within the digestive tract, such as GLP-1 is one hormone that's produced. And so it does have clearly a role in diabetes and metabolism. Dr. Yadav and I, we published several years ago, and now he's an expert in that area. He can address that question a lot more, directly than I can. So, You just have to wait for his talk, which is maybe an hour later.
Yeah. Anybody wants to ask anything?
Yeah, if anybody has anything, I'm there after the meeting. If you guys want, or email me, I'm more than happy to answer any questions or offer suggestions. So thank you.
Just a minute. Dr. Adil is asking.
Yeah.
Just a minute. Dr. Anamika, if a diabetic patients are taking insulin from outside, just a minute. I'll just go. If a diabetic patients are taking insulin from outside, how will the brain work in that situation?
Yeah, very interesting. No, that's a great question. And unfortunately, I do not have an answer for that. I mean, that's a great question because right now we are only able to address what is happening internally in the body with the God-given system that we have, which is the beta cells that are already there. But what the question is amazingly, it's very, very important. So because very advanced diabetic patients do inject insulin, and not just that, we give metformin and a lot of other insulin sensitizing agents or insulin mimetics that change the levels of insulin in the body. How will that work? So just to answer that question directly, what we know and what people think in the field is the neurons in our body, they actually sense glucose. So there are insulin responsive neuron. As you know, insulin signaling is a very important pathway. Every cell in the body has an insulin signaling pathway. Insulin receptor signaling is there in every cell. But what is very important is glucose is the only fuel for the brain. Our bodies can eat everything, but the brain only needs glucose. That's the only fuel that the brain will require. So the brain has a great system of recognizing how much glucose is in your body. So to answer your question about exogenous insulin, so when somebody injects insulin to control their glucose level, they're injecting insulin, but they're actually controlling glucose. So the glucose will go to the brain, and the brain will sense how much glucose is in the system, and the neurons will activate or get suppressed based on the level of glucose in the body. So although I do not have a direct answer to your question because we have not done that experiment, but my expectation is when you give extra insulin into the body, either using intraperitoneal injection or injecting into the brain using ICV, you will change the level of glucose in the body. And the change in the level of glucose will get sensed by the neurons because there are glucose sensing neurons that are in the brain that will sense how much glucose is there for them to live because the neurons need insulin. But that's a great question. I do not have a direct answer to that. But I think it's because insulin controls glucose and glucose is sensed by the brain. That's probably how it will work.
Dr. Adil Zamal from Saudi Arabia. He asked one question. Does COVID-19 cause serious hemolytic disorders? If so, which hemolytic disorder is more common? Does COVID-19 cause serious hemolytic disorders? If so, which hemolytic disorder is more common?
Right. So again, this means I appreciate the question. It's not really my area of expertise, unfortunately. Means if it is anything to do with a diabetic condition where there is increased glucose in the body and that is known to have a lot of other complications that are as a result of the diabetes. In fact, there is now evidence that people who are either diabetic or who are predisposed to type 2 diabetes, they have an increased risk for COVID-19. And one of the reasons for that is the inflammation that goes in the body. So when you have diabetes, because of the systemic diabetes and increase in glucose, your body has a lot of increased inflammation going on in various organs. especially the nerves and the blood vessels. So as a result of that, there is increased inflammation that will be added to by this virus. And as I showed, the virus propagation happens very fast and infects very quickly. So it is possible that some of the conditions such as hemolysis that might be in the context of diabetes may be increased in coronavirus patients, but I don't directly work on those things. So I'm not able to offer any direct answer on that.
Some more questions are there. Varun Chadda wants to ask how the sleeping condition affect diabetes.
Yeah.
Very common.
Yeah. Again, great question. Great question. Means we do not have this, so that's a really good question because the mouse models that we study they are on a daily circadian cycle, 12-hour days, 12-hour nights. So all the experiments that I showed were based on that paradigm. However, we don't agree with that paradigm in daily life. So sleep cycles are different. And there has been studies where circadian rhythm, say suppose you travel from US to India and India to US, and all your circadian rhythm is all messed up. So you're waking up at different times, you're sleeping at different times. That is known to regulate changes in glucose levels. It's because our food habits and our entire system of physiology is changed. So sleep or lack of sleep, all of that plays into your energy state, how much energy you have. So as far as directly how the sleep neurons talk to hunger neurons, it's really understudied. I mean, nobody really has directly studied that. So it is possible though that when you go to sleep and you're activating a certain system in your brain that activates sleep, that region may be talking to some of the regions that are involved in metabolism, such as glucose regulation. But formally, nobody knows really how that interaction happens. But it's a good question. So,
Dr. Sushil, some more queries are there in the chat box. If you don't mind, please give the answers of that queries in that chat box so that we can proceed to the next speaker.
Great. I will do that. Thank you.
On behalf of the organizing committee, we thank you for providing your gracious presence to join us today to enhance our joy and knowledge. Very, very informative session, Dr. Sushil.
Thank you.
Thank you so much. Kindly stop sharing of your slide.
Just stop, right?
Yeah. As we know very well that DNA vaccines can theoretically result in the more long-term production of an antigenic protein when introduced into a relatively non-dividing tissue, such as muscles. Besides, DNA vaccine is relatively inexpensive and easy to produce than conventional vaccines. With high copy origin of replication, good yield of plasmid DNA can be purified from bacteria and suspended in a saline solution prepared for delivery, which may enable timely immunization against emerging infectious diseases. Other advantages of DNA vaccine is that its design and modification is easy. It takes less time for production and formulation and more temperature stable than conventional vaccines. It has a long shelf life and likely does not require a cold chain. School of Biotechnology, Rajiv Gandhi Pradyogiki Vishwavidyalaya, Bhopal, in collaboration with the National Institute of Immunology, New Delhi, has invented 5 recombinant DNA vaccines that are capable of inducing protective antibodies against epsilon toxin of Clostridium perfringens when administered to the animal. It protects against the infection of Clostridium perfringens epsilon toxin. Particularly, this invention provides the membrane-bound cytosolic expression employing LTB epsilon toxin apitopsin, translational fusion with LTB eukaryotic expression of epsilon toxin gene sequence, and their use of effective vaccine compositions. These recombinant DNA subunit vaccines are available to combat epsilon enterotoxemia for veterinary use only. And finally, control mechanisms. These are the 3 patents which were granted to the School of Biotechnology on 26 June 2020 by the Indian Patent Authorities, and 2 more patents are to be granted very soon. School of Biotechnology has filed 1 more patent on 3rd of June 2020 on biopolymer film and method of preparing the same. Moving towards the next technical session, we consider it a great honor to welcome Dr. Lalit Singh Pagbaram, Associate Professor, Department of Ophthalmology, Visual and Anatomical Science, OVAS, Wayne State University School of Medicine, Detroit, USA. He is the man of distinct vision and a fountainhead of illuminating ideas, an idol of knowledge and experience and inspiration to all of us. He is another of our most important collaborators for true science. He will talk about the potentials of gene and cell therapy for diabetes and its complications, use of nucleic acid constructs bearing a TXNIP promoter. Over to you, Dr. Lalit.
So, how do I share this one?
PL sir, I have given you the co-host access. So kindly share your presentation slide.
He's sharing.
Okay.
Can you see or?
Yeah,
yes, sir,
visible.
It's visible.
Visible, and you're audible also.
Okay, so I had a recent medical procedure, so I'm still recovering at home, and my energy level may be a little slower, low then, so please be forgiving. So today I will be talking about a protein called thioredoxin interacting protein. It's called a TXNIP, TXNIP, TXNIP. So this protein is associated with oxidative stress, inflammation, and cell death under hyperglycemic condition. And today, particularly, I'll be talking about the mitochondria and the lysosomal axis. That is the mitochondrial quality control by mitophagy. That is the removal of the damaged mitochondria. We know mitochondria is the energy source, but it also produces a lot of oxidative stress. So we have to have a good control of the mitochondrial quality. So this will be at the end, then our ideas about from this study, how gene and cell therapies can be used for diabetes and its retinal complications. I will talk about diabetic retinopathy here. So diabetic retinopathy is defined by complications of the microvasculature. That's the blood vessels in the eye. So initially, there is some acute and mild complications of the blood vessels, and moderate, where it's called non-proliferative diabetic retinopathy. And as the disease advances, then the blood vessels start leaking, proliferating, and blocking the vision, so it becomes blindness. So recent studies have also shown that these complications start very early by the molecular level, before we see these changes in the eye through the ophthalmic observations. So with the increase in number of people with diabetes or the obesity and other complications in the modern era, the number of diabetic retinopathy, the complication is also going to be increased tremendously in the coming years, especially in the US. It's almost like out of control obesity epidemic. Now, China, India, Brazil, high population countries like Russia, everyone is having all these obesity problems. So the number of diabetes and its complications are increasing. With this comes the burden of the economic burden as well as the familial burdens. So all these things add up eventually. But in the recent times, it has shown that before the microvasculature complications start, that is the blood vessels in the eye start, it is the neuron or the neuronal layers in the eye, because the eye is a part of the central nervous system. It has a made up of neurons for the visual perception, right? So there is a change in the neuronal component in the eye, as you can see the OCT or other imaging systems. There are other complications. There's a night blindness. People with diabetes have a night blindness. And also there's a contrast sensitivity. They cannot distinguish between very light differences in the dark and the light. So in the night, people can get fall down from the stairs because they cannot see the steps. So these kind of things have also been identified early before the complication of the microvasculature. So diabetic retinopathy is both a microvasculature and a neuronal complication. So if you look at the types of diabetes, so it could be a type 1. It is an autoimmune disease because pancreatic beta cells are destroyed. So there is no insulin. So you need to inject insulin throughout your life. So it can happen very early in life. So it's also called juvenile diabetes. And then type 2 diabetes associated with insulin resistance. So obesity and other issues. And then gestational diabetes. Some of the ladies, females, during the pregnancy, they develop this insulin resistance and they can also cause some diabetes, but they do return after the childbirth. So whether it is type 1, type 2, or gestational, there is a high glucose level in the blood and the tissues, and that can cause a lot of complications if it's prolonged. So one of the genes that is strongly induced or highest level induced at the highest level, both in the beta cells, heart, muscle, eye, kidney, all these tissues is called a TXNIP, thioreducin interacting protein. So this protein is a oxidative stress inducer, and it causes inflammation and premature cell death. So at the end, it leads to the complications of the retinopathy, eye, nephropathy, neuropathy, and all sorts of complications in the system. So diabetic retinopathy in type 1 or type 2, some form of retinopathy will develop in diabetes. If you have 10 to 15 years of diabetes, either retinopathy or nephropathy, neuropathy, some form of complication comes. So in terms of the metabolic dysregulation, hyperglycemia cause this dysregulation of many metabolic pathways, including hexosamin pathway. This is the UDP gluconate or glycosylation pathway, alteration in the glycosylations, polyol pathway, fructose sorbitol, PKC pathway, ACE and RACE advanced glycation products, and the mitochondrial stress pathway, lack of bioenergetics and energy and the oxidative stress production, mitochondrial. So in our lab, we are interested in two of the pathways. That's the hexosamin pathway and the mitochondrial stress pathway. So we use a type 1 model of diabetes using streptozotocin, which destroys the pancreatic beta cells. So there is no insulin production. It's like a type 1 model. So if these animals are maintained, we maintain say 4 weeks to 8 weeks, depending on the experimental conditions, we see that this protein, TXNIP, that's the thyroreducin interacting or inhibiting protein. By name, it interacts with thyroreducin. Thyroreducin is an antioxidant and survival protein. So it has a significant increase in the diabetic animals in the retina. So after 4 weeks or 8 weeks, we sacrifice the animals and harvest the retina and the process for different kinds for the mRNA, protein, or histology, histosections. So from this, it is clearly seen that in the retina, in diabetes, TXNIP is increased significantly in the mRNA and the protein level, and as well as in the histosections. So this red is the TXNIP. So in all layers of the retina, so retina has several distinct layers, nerve layers and the photoreceptor layers. So my mouse is not working. So in any case, this TXNIP expression is associated with changes in the retina, that is thickening of the basement membrane. This fibronectin is one of the proteins for the basement membrane. So showing that the blood vessels get a thickened basement membrane is a response to the blood vessel injury. And then in the ganglion cell layer, my mouse is not working, so I'll just go through one by one. So if you look at the case phase 3 staining, here we see this case phase 3 is increase in the diabetic retina in the ganglion cell layer. That is the one of the neuronal layers, suggesting some neuronal cell death or injury may occur. And the GFAP, that is the glial fibrillary acidic protein, that radial expression is increased, the green ones. This is the glial cells that helps support the neurons in the retina. So there's a reaction to the injury to the neurons. So it's called a gliosis. So gliosis is a way to protect the neurons, but if it continues for a long time, then it becomes detrimental. It can injure the neurons itself. So that's how we saw in the beginning. So how does this TXNIP work? What mechanism is it? Is that it is a protein strongly induced by diabetes and hyperglycemia in pancreatic beta cells, retinal cells, in all the cells that use glucose. So it binds to the thyroreducin in its redox active site at 32, this cysteine. It can cross-link with the 247, the cysteine. So it can block the activity of thyroreducin. So once you block the activity of thyroreducin, the cells tissue becomes oxidative stress, inflammation, and the cell death. So retina, being a fully differentiated cell tissue, if the cells die, you cannot be replaced. So it's neurons, if they die, they cannot be replaced. So there will be a functional deficiency. So in the cellular system, you have thyroreducin 1 and thyroreducin reductase 1 systems. They work to maintain the reduced state of the cells. And thyroreducin 1 is present in the cytosol and nucleus. And the thyroreducin 2 and the thyroreducin reductase 2 are present in the mitochondria. And TXNIP is present in all compartments of the cell, including mitochondria, nucleus, and cytosol. So it can block the function of this thioreduction 1 and 2 in all these compartments, and ultimately lead to oxidative stress, inflammation, cell death, and neuroinflammation. So complications can start, including diabetic retinopathy. That's the model of how thioreduction interacting protein cause cellular defects. So in this, we show that the number A, so 1A, here we show is that in the diabetic retina, there's an increase in the glycosylation process. And this can be blocked by azathioprine is a compound that inhibits the glycosylation. And on the right side, you have that compared to the normal retina, the diabetic retina has TXNIP expression, the red color. But when you inject the azathioprine, which is inhibitor of the hexosamin pathway, then you block the TXNIP expression. So hexosamin pathway is involved in the expression of TXNIP. And if you look at the number C here, the TXNIP is downregulated in the mRNA level, and the COX-2 is the cyclooxygenase 2, that is the inflammatory enzyme, is also increased in diabetes, but azathioprine can block it. Fibronectin is increased, it is downregulated. So hexosamin pathway and the TXNIP are involved in some of the molecular defects early in the process of diabetic retinopathy. So similarly, if we inject in the retina, in the vitreous, because of the blood retinal barrier, so systemic drug or oral delivery doesn't work in the eye. So you have to inject directly into the eye through the vitreous. So that's the way it is. So if we inject the siRNA to the TXNIP, we can knock down TXNIP and then the expression of COX-2 and fibronectin, suggesting that TXNIP expression is upstream of all these molecular defects. Similarly, in the D, down, you will see also this GFAP, that is a glial fibrillary acidic protein, is also enhanced radial expression, but with the siRNA, it is also reduced. So we call this siRNA direct, called PTGS. Post-transcriptional gene silencing targeting the mRNA is called PTGS. So similarly, if you look at the inflammatory molecules, like the IL-1 beta, INOS, and then the receptors, inflammatory receptors like TLR4, P2X7R, all are increased in the diabetic retina. And also IL-1 beta is increased, but if you inject the TXNIP silencing, here we use a TZS, that's called a transcriptional gene silencing in the B, panel B. So, and also GFAP in C is also reduced by this TXNIP TZS. So the TZS transcriptional gene silencing is a method to close the promoter of the TXNIP gene by these double-stranded siRNAs. It's a transcriptional method, not the mRNA method. So in this stress, does the mitochondrial stress or neuronal injury occur? So we look at the mitochondrial enzymes and proteins. That's a BDEC. This is the membrane protein is increased. And then in the C, the aconitase 2, that is a TCA cycle enzyme, is increased. D, we have the heme oxygenase is increased, suggesting that mitochondrial stress and response is occurring in these retinal cells. So along with that, there's a neuronal injury because if you look at the E and the F panel, that tyrosine hydroxylase TH is down, as well as synaptopodin, one of the synaptic proteins, is also down. So TH is the tyrosine hydroxylase is an enzyme that in dopamine synthesis. So there is a neuronal injury in this early diabetes. And does it induce the removal of these injured molecules or any autophagic process or mitophagy? That's the ATZ5, one of the autophagy genes, is upregulated along with the heat shock proteins. So there is some response to the injury in the retina. So ATZ5 is one of the important genes or proteins for the autophagy and mitophagy. So as mentioned here also, A, B, and C, Sniff is upregulated in the diabetes. And along with this, in the D, staining also shows red Sniff is increased, and siRNA can block the expression. So in the E, if you look at this, in the middle panel, diabetes scramble RNA, you have a lot of this LC3B red punctate. There are so many punctates. These dots. These are the autophagophores or autophagy bodies. And if you have a siRNA Sniff, then it can reduce some of these autophagic processes. And as a demonstration, again, the GFAP, the green filaments are also reduced. So Sniff is involved in some of these other mitophagic or autophagic processes. So as mentioned, the retina is a very well-organized neuronal component of the brain. It has a vitreous in the eyeball where the light comes in, and then you have the ganglion cells, GC, and bipolar cells, and then photoreceptor cells. So even though the light comes from the vitreous, the photoreceptors, light receptors, are at the opposite side first. So the photoreceptors get the light, capture the light, a signal goes to the bipolars, and then the ganglion cells, and all the filaments go to the brain for processing, right? So in our case, we'll be focusing on two of the supporting cells, that is called the MC, that is the MC is the Mueller cells, glial cells that support the neurons. It expands the entire length of the neurons, retinal neuron, and its function is to remove the glutamate from toxic, like the neurotransmitter glutamate, to uptake and then convert it to glutamine for further action, and also secretes a lot of the growth factors for the neurons, and then metabolic support. So it plays an important role. That's what we see, the green filaments in the previous slides. That is from these Mueller cells. So it's a reaction to the neuronal injury. And another cell type is the RPE, retinal pigment epithelium, at the base of the photoreceptors, these cells. So the RPE supplies glucose, oxygen, and other nutrients to the photoreceptors, and also has a pigment, so excess light can be reabsorbed. It also removes the outer segment of the photoreceptors, because each day when you have this light processing, that damages all these photoreceptors and the membranes, outer segments, these these segments, they are embedded into this. So it has to be renewed every day in the night. So in the daytime, we use the visual, but they get damaged. So in the night, it has to be regenerated again. So it is a very, very active process that is done by these RPE cells to phagocytosize all these outer segments and then degrade in the lysosomes. So this is a very active cell type. So it involves a lot of the phagocytic activity and the support to the neurons. So any injury or any damage to the RPE cause damage to the photoreceptors and the visual defects, like in the AMD or the age-related macular degeneration, retinitis pigmentosa, blindness. All these involve the defects in the RPE. So even though the retina is a very small organ, it consumes the most glucose and oxygen in the body because it's a very, very active, metabolically active tissue. So by that, it also means that its mitochondria is very active in producing the ATP for the visual function. So mitochondria, when it produces ATP in the electron transport chains, it releases a lot of the radicals, oxygen radicals. So it continuously damages the mitochondrial membrane, protein, and DNA, all those components. So these damaged mitochondria, they cannot produce ATP, but they continue to produce the reactive oxygen species and the toxic radicals accumulate. So they need to be isolated or fission. So in the mitochondria is a very long filamentous process, and it doesn't damage all of them at the same time. So it damages part of it. So the parts that are damaged are isolated by fission, and then they are marked by ubiquitination, and they engulfed by a double membrane autophagophore and combined with the lysosomes to form the autolysosomes. And then they are digested for recycling the components. So as the old or damaged mitochondria are damaged and removed, new ones need to be make mitogenesis or making of the new ones, and then they will fuse by fusion so that the number of mitochondria can be maintained and energy ATP level can be maintained. However, in diabetes and the age-related diseases, the cycle of this mitochondria to lysosomal flux is dysregulated. So if this mitophagic flux is too slow, then damaged mitochondria will accumulate and the radicals will be accumulating. And if it is too fast, damage is coming, and if it is too fast to remove the mitochondria, then the mitogenesis may not be able to follow up. So you have a less number of mitochondria and less ATP production. So when you have these lysosomes are accumulated with many of these damages, like proteins, aggregates, or the organelles, mitochondria, or other organelles are accumulated, the membrane becomes leakage, destabilized, LMP, or lysosome membrane permeabilization, and can leak out the components, and it can cause inflammatory processes like inflammations, NLRP3 inflammations, and then neurodegeneration or neuroinflammation. These kind of things can propagate. So maintaining the mitochondrial quality control by mitophagy as well as mitogenesis is important. So that's why we look at some of the parameters in two of the cells, the Mueller cells and the RPE cells. These are the supporting cells for the neuron. So we We use a cell line called RMC1, that is the Mueller cell line. So if you expose these cells to the high glucose conditions for 5 days, we use many different days and found 5 days is optimal for these kind of experiments. So we can see that Sniff expression is increased in the high glucose, in the total extract, as well as in the mitochondrial fractions, like in the D, panel D. So Sniff expression is increased in mitochondrial itself, and it can cause oxidative stress, mitochondrial stress by mitoshocks. So there is a mitochondrial stress, and then the membrane depolarization occurs as a JC1 probe shows that one. So there is a mitochondrial damage when you expose these cells to the hyperglycemia. So to further understand the role of the Sniff, we use a CRISPR-Cas9 method to knock out Sniff in these cells. So in this process, it's very common nowadays, very easy to do it. So in this process, we targeted the exon 1 of the Sniff gene, where you have a PAM sequence and all those things. So we characterized and we show that with this process, we can knock out Sniff in these cells. So I'm just going to leave it there. So we use a CRISPR-Cas9 to knock out Sniff in these RMC1 cells. So it's a complicated slide, but what this shows is that in a mitochondrial fraction, if we isolate the mitochondrial fractions from these cells after treatment with high glucose, there is an enhanced level of the DRP1. DRP1 is the dynamine-related protein that involved in the fission or fragmentation of the damaged mitochondria. And then down in E is the parkin is also increased in the mitochondria. And parkin is a protein that ubiquitinates the damaged mitochondria that marks for degradation by lysosomal pathways. So by doing double immunostaining, we saw a lot of these combinations of these mitochondrial protein COX-4 and the parkin or DRP1 or ubiquitin or optinurin. A lot of combinations show that there is a mitophagic process in the RMC1 under high glucose conditions. And if we use some of the double membrane proteins, the LC3B is a double membrane autophagophore marker, and then LAMP2A is the lysosome membrane protein. So they are co-localized, suggesting that these damaged mitochondria are targeting to the lysosomes by yellow co-localization in panels B down in the insects. You can see some yellow co-localizations. But if you knock down Sniff, these yellow co-localizations are not observed, suggesting that mitophagic flux is reduced and mitochondria are more filamentous than breaking. Similarly, also we look at the RPE cells, what happens with the hyperglycemia in 5 days. There is a Sniff increase, and then mitochondrial JC1 is down in C, suggesting depolarization. ATP levels are down, so energy level is down. And cell live and dead is reduced, suggesting cell death occurs. And NAC, N-acetylcysteine, that is the antioxidant, can block some of these cell death, suggesting there is a oxidative stress involvement. And also the mitochondrial enzyme COX-4 is colocalized with the LAMP2A, that is the lysosomal protein, suggesting there is a mitophagic flux, mitochondrial fraction going to the lysosomes. So under high glucose condition, you will see a colocalizations with yellow processes in RP cells. So we also use the mitochondrial targeted GFP to see whether mitochondrial fragmentation occurs. And there is a fragmentation of the mitochondria in C. We'll see panel C, that under high glucose, mitochondria are seen as small dots. And in the A, we'll also show that lysosomes, when we use M-Cherry, LAMP1 M-Cherry lysosomal probe, there is an enlargement of the lysosomes. So if you look at the size of the lysosomes in the high glucose, is much more bigger than the low glucose conditions. It is 2-3 times bigger, suggesting that there is a flux of these damaged mitochondria to lysosomes, and they are fusing together, but they are unable to process or excrete out the products. So high glucose can do damage to the mitochondria and also lysosomal degradation pathway. So similarly, we use one of the compounds called M-lexanox. So M-lexanox is an interesting compound because it is used as a ointment for canker sores in the mouth. And recently also it has been tested for treatment for obesity in the mouse in the University of Michigan scientists. So M-lexanox has been shown to prevent some obesity in the mouse. And what does M-lexanox does is it is the inhibitor of a protein called TBK1. There's a tank binding protein kinase 1, and it phosphorylates the mitophagic adapters. That's optinurin and P62s. Those are the proteins that enhance the mitophagic process. So in our case, when you treat these cells with high glucose and M-lexanox, it can slow down the process of degradation of this optinurin and the P62. That's the adapters for mitophagy. So in the low glucose and high glucose, their levels are low because they are going to the lysosome and they are degrading. Whereas if you have the M-lexanox, that is slowing down the process of the degradation of the optinurin and P62. So this compound has been suggested to have some effect on the eye diseases, including the glaucoma and the other complications. So we use that one to see what happens to the lysosomal enlargement. So if in the presence of M-lexanox, we do not see the enlargement of the lysosomes. And similarly, N-acetylcysteine antioxidants can also reduce the size of the lysosomes, suggesting this mitophagic flux is the one that is causing the size. So next, we also try to monitor the mitophagic flux directly. We use a probe called mitochema. So mitochema is a chorale protein. So when we express them in the mitochondria, it gives you a green color at the alkaline pH. And if it goes to the lysosomes via mitophagic flux, then the pH becomes 4 to 5 acidic, then the color changes to red. So if it is green, it is in the mitochondria. If it is red, it is in the lysosome. So the single probe can be used to see whether mitophagic flux is there or not. So as a proof, we use a CCCP, that is to make the mitochondrial membrane depolarization and undergo mitophagy. So you will see in the second panel, CCCP, a lot of them have a red color. So suggesting that that's going to the lysosomes compared to the first panel, that's the control, all are green. So this probe is a useful method to monitor mitophagy, but this is done only in the live cell imaging, because if you fix the cells, then the pH will be changed. So similarly, using this probe, we can also show that mitochondrial filamentous or mitochondrial have a long filaments in the low glucose conditions, but under high glucose, their filaments are broken, and you see a lot of the red color in the high glucose panel, suggesting there is a mitophagic flux. So which can be blocked by amylexanox or N-acetyl antioxidant, or by BFA, that is the blocking the lysosomal fusion. So this probe is quite useful to study mitophagic flux directly without any staining or without any colocalization or anything. Single probe can do it. So similarly, if we knock down Sniff, so here you have high glucose, you have a lot of red in this, but if you knock down Sniff, in the second panel C, you don't see all those red mitochema, suggesting that it's blocking the mitophagic flux. So from this, what it says is Sniff expression is involved in the downstream mitochondrial stress and mitophagic flux processes. And we don't know what it is. Mitophagy is a defense mechanism for the cells to survive, like autophagy. However, in the disease process, it can be slowed down or it can be activated, accelerated. So if it is slowed down, all the damaged mitochondria will be accumulated and producing a lot of the oxidative stress markers, and then components, mitochondrial components released into the cytosol. They are inflammatory. If it is too fast, you are removing the mitochondria and there is a bioenergetics failure and the lysosome damage. So at any stage of the disease, what is the optimal process of mitophagy is still not known. So it is a question by many, is mitophagy good or bad? So depending on the context, it is good or it can be bad as well. So in this process, summarize is that in diabetes hyperglycemia, Sniff expression is increased, and it can block the thyroidexin 1 and 2, cause oxidative stress, mitochondrial stress, and mitophagic flux. So if it is too much flux, lysosomes get damaged, and it can cause, again, inflammatory processes and disease process. So that's the brief process from Sniff. So we think that it is the early process that Sniff is the one that is causing all these downstreams. So now coming back to the gene therapy approach and the things from these studies we have done for the Sniff, many others. manuscripts we have shown is that the Sniff promoter is activated by glucose metabolites, glucose 6-phosphate, fructose 6-phosphate, glucosamine 6-phosphates, and it has the E box, and the transcription factors that bind to them are MONDO-A, MLX, carbohydrate response element binding proteins, and these proteins, transcription factors, combined with the P300 head histone acetylase, can open up the promoter and for gene expression. So Sniff gene expression is inhibited by insulin, but in the type 1 or type 2 diabetes, when there is no insulin or resistance, then hyperglycemia prevails and the Sniff gene expression is increased. So as long as there is hyperglycemia, Sniff gene is induced. And if you remove the hyperglycemia, then Sniff expression is reduced, even if you don't have insulin. So we thought that Sniff promoter, this promoter can be exploited to induce gene expression of our interest, okay, under hyperglycemic condition or diabetic conditions. So if we link this Sniff promoter to either an antioxidant gene, inflammatory gene, or redox proteins like TRX1, cDNA, it will be expressed under hyperglycemic condition, but it will be at the basal level under the low glucose or normal glucose. So this promoter can be controlled by a physiological molecule, that is glucose. So to check this idea works, we use a Sniff promoter and a link to the thyroreducin 1 and express in ARP1. So in this case, if under normal glucose, low glucose, or high glucose in the control gene vector expression, there is no change in the level of TRX1 or TRX2, but in the construct promoter, Sniff promoter and the TRX1 construct, then high glucose can increase TRX1, and interestingly, Sniff also is reduced. So by this construct, single construct, we are able to achieve two aims. One is to increase the antioxidant, and another is to decrease the pro-oxidant itself. So, and a lot of other signals, low glucose, high glucose, like other reporters can also be induced by histone acetylation and hyperglycemia. So this construct works under high glucose or histone acetylation. So similarly in the RMC1, also it can work, and it can also control some of the mitophagic processes in this construct with the Sniff promoter and TRX1. So in theory, in an in vitro system, cell system, this gene construct works under hyperglycemia, and it can be a good platform to deliver genes of interest. So in our idea that in vivo translation in the diabetic retinopathy, so we can construct the vectors AAV2 or the viral delivery systems, use a Sniff promoter and the gene of interest, TRX1, or the neurotrophic factors like GDNF. And these constructs can be injected into the retina into the vitreous directly, and they will incorporate into Mueller cells or some other neurons, and they can protect some inflammatory or neurodegeneration in early diabetes. So that's one idea. Or ex vivo, these constructs can be introduced in the autologous stem cells or IPS. And in our case, we use the adipose stem cells, adipose derived stem cells, ASCs. So when we use these cells with the transfected with the trypsin promoter and insulin gene or adiponectin, they can also produce these genes. So particularly, we are interested to see whether these adipose stem cells, when we manipulate the genes, can they produce insulin under high glucose conditions, and can we make them secrete so that it can be transplanted subcutaneously, because adipose tissues are subcutaneous cells, and they survive very, very long. So one of the failures for beta cell transplant so far by many labs and the industry is that beta cells don't survive outside their pancreatic environment. So transplant has to be done very frequently. Either you put encapsulated system under the skin. So far it hasn't been successful. But if we can make some of the non-beta cells to produce insulin and make them secrete, then that will help to control the blood glucose after each meal. So because these adipose stem cells and iPSCs, they are under the influence of GLUT1, that is the glucose transporter 1, whereas the pancreatic beta cells is a GLUT2. So in the GLUT2 beta cells, you need a high level of glucose to induce insulin production. Whereas in the GLUT1 with the trypsin promoter, you don't need that high level of glucose to induce the expression of insulin. But whether these adipose stem cells or other cell types, can they process and secrete insulin? That is the question that we and many other labs and the companies are asking. So that's where we are. So I'm going to stop here. Some of the lab members and collaborators here. Thank you very much. So I stop here.
So very nice. Can I ask a question?
Sure, sure.
Yeah. So very nice. Pretty informative, and ties in, glucose and a lot of the glucose and insulin responsive pathways to what's happening in the eye, and of course all the degeneration that happens as a result of glycemia. So the question I have is about the oxygen itself. So now, have you tested whether the expression of this gene or any of this pathway with relation to hypoxic conditions, and how does hypoxia modulate any one of these, either the promoter or any one of these transcription factors that then later on progress to full-blown disease? Means, what is the role of oxygenation here?
Yeah, hypoxia can be developed at the end, towards the end of the complications, because blood vessels are not perfusing well. So if there is no hyperglycemia, hypoxia will shut down TXNIP, because TXNIP blocks glucose uptake of the GLUT1 and GLUT4. So hypoxia needs more glucose to get in, because in that case you have the glycolysis, anaerobic respiration to get the ATP. So it switches from mitochondria to glycolysis for ATP production, which means you need a lot more glucose to get in. So initially it will shut down Sniff so that more glucose can come in. But as more glucose come in, then Sniff will start coming up. So you will have balance. But under hyperglycemia and hypoxia, Sniff will be upregulated. Because as long as there's hyperglycemia, nothing can block Sniff. Even insulin cannot. See, insulin or IGF-1 will block Sniff under hyperglycemia. But the effect, it lasts only about 4 hours. After that, it will shoot up again even higher. So that's why we thought that this Sniff promoter is under the control of glucose. That's a physiological molecule response. So if you have a gene of interest linked to this promoter, then under hyperglycemic condition, your gene will be expressed. And under physiological control, then the gene will not be expressed. So you have a control. It's like saying, okay, take your pill twice a day after meals. So after meals, your gene will be expressed when high glucose is there. And then physiologically, so in the interest of these adipose stem cells, because in obesity, obese people, you have access to the adipose tissue very easily, liposuctions, and they can be manipulated ex vivo outside and then transplant it back. So it's autologous. There will be no rejection or things. And these adipose tissues, they survive very long time, so you don't have to inject very frequently. So that is the idea. That's why we went to the adipose stem cells, because obviously that adipokines, because in the obese and the diabetic, type 2 diabetes, adipokine level is lower. So it can be linked to the Sniff promoter and enhance its expression under hyperglycemic conditions. So those are some of the thinking.
Karan has asked one question. How does diabetes, hypertension patients are more vulnerable to COVID-19, and how does these patients' bodies respond to infection? I'm repeating, how does diabetes, hypertension patient are more vulnerable to COVID-19, vulnerable to COVID-19, and how does these patients' bodies respond to infection?
Yes, because diabetes, obesity, hypertension, these are three things going to go together. So I had a seminar a couple of years back, I called Diabetes, instead of diabetes, THY. So obesity, diabetes, and hypertension together. So oxidative stress, inflammation, these are occurring in these systems. And so therefore, as we talk about COVID or any other inflammation or infection, there's less protective mechanism in the body. So any diabetes or obese people have a high rate of infection and recovery, slow recovery than normal people. So hypertension is something that people don't talk about or doesn't give together with all these other two, obesity and diabetes, because obesity society, diabetes society, hypertension society, they are all separate organizations, and it seems they don't talk to each other quite often. So everyone has their own guidelines. But to a person who is type 2 diabetes, there is going to be hypertension as well.
Another question from Dr. Adil Zamal from Saudi Arabia. Keeping in view the current scenario, do you think CRISPR application generates more effective and reliable results in case of diabetes, retinopathy as compared to classical approach of siRNA? Which one yields stable expression at translational level?
Now, CRISPR is a knockout process. So diabetes is a multi-gene process. It's not a mutation or it's not a single gene mutation disease. So CRISPR's knockout is not a very good for diabetes. It's good for studies, but as a therapeutic process, if you knock it out, because most of the genes are essential, so you don't want to knock it out completely. So you need to keep them in the basal level. So in the case, knockdown is better than the knockout for diabetes. But CRISPR also can, instead of knockout, CRISPR also can use for knock-in, like D-Gas9 or deactivated Cas9 can also introduce genes. So it's a very versatile technology. So it can be used in different ways. And CRISPR is good for knocking out the mutant genes in the diseases where you have a single gene mutation, and you can knock out those genes and then put a new one, knock in. For example, in the eye, if you have a retinitis pigmentosa, you can knock out those genes that are defective and then knock in those good ones. So gene therapy so far is approved for the eye disease, mutational diseases like the retinitis pigmentosa, because gene defect, whereas insulin resistance is not a mutation, it's a multi-gene metabolic process. So we don't know which one to knock out or which one to knock in. So it is more, gene knockdown is better.
Another question from Dr. Sonam Chawla. Excellent presentation, sir. I was curious to know if you intend to explore any specific pathways for aged animals. We know that elderly have higher incidence of retinopathy. So any idea to explore telomerase or Sartorus?
Yeah, like any other gene disease, diabetic retinopathy involves a lot of cell types in the retina, a lot of the genes. So we don't know which genes to exactly to target. So in our case, we propose is Sniff is an early response gene. It is expressed as long as hyperglycemia is there. So it doesn't matter at what stage of your disease, whether it's the beginning or the middle or the late stage, Sniff is always expressed in the hyperglycemic conditions that can cause oxidative stress and inflammation and cell death. So this is one of the good targets, because in the gene expressions change as the disease progresses. So if you knock out or knock down one gene in the early, that may not be important in the late stage. So depending on the disease state, we have to identify is stage-specific gene expression. Because when you go to the clinic, you don't know the person has been in diabetes for 5 years, 10 years, or 15 years, or 20 years. And the doctor gives you the same drug, but it may not work for all of them, depending on the stage of the disease progression. So those are the ones that we have to work out, is at what stage specific genes are changed, altered. And those specific genes has to be targeted based on the personalized disease process. That's what we think.
I think if anybody wants to ask more, kindly write in the chat box and Dr. Lalit will answer that. If I'm not wrong.
That's it.
Excellent session, Dr. Lalit. Thanks a lot for enlightening and updated us regarding one of the aspects of T2DR with TechSnap promoter. Dr. Lalit, please stop sharing your slide. We sincerely thank you to make this day an occasion we will always remember. Memorandum of Understanding between Rajiv Gandhi Pradyogiki Vishwavidyalaya Bhopal and Madhya Pradesh Council of Science and Technology Bhopal on the basis of the objectives of the agreement are to establish a mutually beneficial relationship built on academic and scientific cooperation. Areas of cooperation include the exchange of ideas on research, publications, academic programs, and research projects. Initially, this MOU was executed on 21st of July 2020, but due to the present conditions of the pandemic in Bhopal, it will be executed shortly. Now, we are proceeding towards the third technical session, under which we call upon none other than our distinguished speaker, Dr. Hariom Yadav. He is an assistant professor in molecular medicine in Wake Forest School of Medicine, Winston-Salem, USA, to address the gathering and to give his precious inputs to all of us. He is going to discuss on gut microbiota and its modulators in diabetes and obesity. Dr. Yadav, over to you. Dr. Yadav, am I audible? Yes.
Can you guys hear me here?
Yeah.
Okay. So I would like to first thank you for the organizers for inviting me here. And I'm really grateful to share this stage with my mentor here, Dr. Sushil Rane. So thank you, sir, for, for joining here. I'm really fortunate to present here and share some of the work what we are doing here, and also some of the work which I did with Dr. Rane in NIH. I will be showing here. So really great. Let me just play out. Now I'm not sure whether you guys can see my cursor also here moving.
Yes, everything is visible.
Great. So I will be talking a little bit about how does the microbiome, like especially living in the gut, impact our metabolic health, which is especially diabetes and obesity. First of all, just for formality, I have to give this disclaimer. I'm associated with these companies here for funding wise as well as working wise. Now looking on the overview of my talk, I will try to make my talk to like more general, not going much depth in the signaling pathways in the cellular, but I will try to relate our daily life, how really we can relate this science to our daily ongoing things, eating behaviors, as well as how does that affect our metabolic function. So mainly I will be talking on three topics. One is gut-brain axis to regulate food intake. It's kind of the concept when nobody really tells us when we are hungry. Nobody tells us when we should stop when we are eating. So how does that really get communicated between gut and brain? And how does that plays the role in obesity and diabetes? Because obesity is a condition where it depends on how much we eat. and how much we spend as energy expenditure. So the more we eat, the more we can gain the weight. The another thing is gut, the microbiome and butyrate and leaky gut condition, which is very common in the older people. So I will little bit talk about that. And then whether the gut microbiome signature can be used as a biomarker for different human diseases. And this is kind of the focus of my labs, these three questions we are trying to work on. Now, when we talk about the human microbiome, in reality, actually, we are more microbial than the human. The reason is because we have 10 times more cells than our own human cells in our body. And although these cells are more tiny, so they are less visible, but in numbers, they are more. If we talk about like looking on the other way, who regulates whom, actually the microbiome can hijack our body cells. So one of the reason is, you can simply say is, in the human genome, only 23 to 24,000 genes are expressed. That means 23 to 24,000 proteins are made by our own cells. However, in our body, inside or outside, more than 300,000 proteins are made by the bacteria, and they get secreted and get imposed on our cells. So you can see by numbers how many times the more proteins are made by microbiome than our own cells. Now we talk about the majority part of the microbiome. Although the microbiome is present in different sites of the body, like our whole skin is full of the microbes, our eyes, our mouth, ears, you name the most of the sites which are exposed to the environment has the microbiome, but the gut is or like our colon is the full of the microbiome. And in general, if you talk about, there are like 2 kg of the bacteria we every day carry a normal person. So you can see like how much body mass is made by the microbiome itself. So our colon itself has 100 trillion bacteria living in, and which is like 10 times higher than our own cells made the whole body. Now, when we talk about the microbiome itself, although the microbes have been always there, but they were difficult to study earlier, but new technologies came, and especially this next generation sequencing, now we can measure the microbiome composition. However, 98% of the bacteria, we still cannot grow them in the lab. But by doing this DNA sequencing, we can find out who there is. So when we look on the microbiome, and that's how like you cannot estimate the exact number of the bacteria, but you can measure the proportion, what kind of bacteria and what proportion they are living in, and that they are always presented as a percentage form. So if we look at typical microbiome signature here, you can see this is the gut microbiome. There are 2 major types of phyla, Firmicutes and Bacteroidetes are mainly make the whole microbiome in the gut. But then there are, so 80 to 90% is Bacteroidetes and Firmicutes. And then there are other bacteria called Actinobacteria, Proteobacteria, Fusobacteria, Verrucomicrobia, 10 to 15% maybe. And this is, I'm talking about when we talk about the phylogeny of the bacteria. So bacteria, the microbiome is always presented in the form of the phylogeny. So like if you start from the bacterial kingdom, then the phyla, family, genera, and species, that kind of distributions there. Now, when we talk about the microbiome in the human health, you name any disease related to human health, you will find the correlation or the relationship of the microbiome. And some of them you can see here, they are listed here. My lab is mainly focused on, as I said, the gut-brain axis, looking like we are also looking on the Alzheimer's disease and the food intake dynamic by looking on how the gut and brain, they communicate each other. And looking these impacts on the aging, obesity, and diabetes related conditions. Now, when I talk about is what specifically we are looking is, we are looking the microbiome and leaky gut and inflammation interaction, what is happening in the gut. And there are some mechanisms are involved in the short chain fatty acid, which are produced by the microbiome. And these short chain fatty acids are the major metabolites. One of them is butyrate, which works through the signaling pathways called the FFAR2 and FFAR3. These are the free fatty acid receptors 2 and 3. And they are mainly expressed in our human cells as well as the mammalian cells. And they sense what is produced by the microbiome and impact the leaky gut and inflammation, and impact the different kind of cells like the L cells. These are the intestinal endocrine cells, produce the important gut hormones, which play the role in the gut and brain communication, how much we will eat and how our body will respond to particular diet, as well as the goblet cells. These goblet cells are the one which produce the mucus that maintains the integrity in our gut to reduce the leakiness. Otherwise, anything what we eat will go in our blood. So, but there is a selective absorption happens in the intestine, and how does that integrity is maintained to the gut is important. So I will go first with the gut-brain axis, microbiome gut-brain axis to regulate the food intake. As I said, the microbiome has been related to any kind of the human disease. Now the question comes always is it just the relationship or the microbiome cause this disease? Because any health condition can change the microbiome. So these are the first studies came back in 2006, and those are the kind of pioneering studies came into whole microbiome field and kind of blown up this as the whole area. And the study is microbiome in case of obesity. It's not just the microbiome is different in the obese person versus the normal person. Actually, microbiome can cause the obesity. And this is the classical example experiment was done is when they took the microbiome from the obese twin versus the lean twin. Why they choose the twins is because genetics plays the role into obesity dynamics. So to exclude the genetic component, they choose the twins. These are homozygous twins. One group is obese, another group is lean, and transplant this microbiome to the germ-free mice. And these germ-free, and maintain these germ-free mice eating the same diet, same environment. And you can see here, the mice which got the microbiome from obese twins develop the obesity or adiposity, whereas the mouse which got the lean microbiome remain lean. So you can say that is if you transplant the microbiome from obese to a normal person or normal mouse can actually increase the obesity propensity itself. So it's a causative agent, it's not just the relationship there. And then the question comes is how does the microbiome can increase the obesity there? So there was another following study from the same group, Jeffrey Gordon. What they did is when they take the germ-free mice, there is no microbiome in these mice, and feed them on high-fat diet, these mice remain lean, they don't gain weight. So what they are saying is the microbiome presence itself makes the obesity susceptibility high. And if they take the normal microbiome and put in the germ-free mice and feed the same high-fat diet, these mice start gaining the weight. So that shows that if the microbiome, even the normal microbiome is there, and that is put on the high-fat diet will increase the obesity. One of the mechanisms what they said is when the microbes are there, so whatever we eat, it will get absorbed, whatever our body will digest it. The rest of that will pass out as a stool. But if the microbes are there, microbes will further ferment, which our body cannot digest. And that fermentation end products will absorb further in our body, will be kind of extra calories will be harvested from our food. And that's how that extra calories will become in long term accumulated and cause the obesity there. So having the microbiome itself is a risk, but reality is nobody can be germ-free in our real scenario. So, but that's kind of the mechanistic studies are done. So another question comes is if the microbiome itself is posing the risk of obesity, then there is another component called the probiotics have been used, and kind of their use is increasing day by day in the normal society, that they are the risk for developing the obesity. Because if the microbiome itself is there, probiotics are the part of the microbiome, they might be promoting the obesity there. So the increasing rate of the obesity has to be related with the something, and whether the probiotics are linked there. So this was a Nature Microbiology, Nature Reviews Microbiology editorial was there, and they posed the question is whether the probiotics are good or bad for obesity. By taking the one example, as I showed in the earlier slide, is presence of the microbiome itself increases the risk of obesity. Another thing is they took the example of the animals which are fed on the probiotics diet gain the more weight. In the meat industry, especially, probiotics has been used as a growth promoter. So by taking these two evidence, and then they argued is no, really can somebody can prove that whether the probiotics are good or bad for obesity and diabetes. To answer this very broad question, what we did is we choose the 5 probiotics which are commonly available into the US, most of the pharmacies, with really no contacts or no help of any companies. This was purely scientific studies, which I'm fortunate Dr. Rane have allowed me to carry on that one. What we did is we choose the mouse, control mouse, the normal chow fed mouse, as well as the only high fat diet fed mouse, and we feed these probiotics on the high fat along with the high fat diet, and see that whether any of these probiotics will promote or decrease the obesity among these mouse groups. What interestingly we found is none of the probiotics what we selected actually increased the obesity or body weight gain on high fat diet. Some of them were less effective to suppress the body weight gain, and some of them were very effective. Like one of them was the VSL3, you can see here, was very effective to keep the body weight as similar as the mouse which are eating the normal chow, suggesting that some of the probiotics first of all, none of the probiotics are bad from these selected 5. Another thing is some of the probiotics can be beneficial to protect the high-fat diet-induced obesity. Then, so we call this study as a preventive study. This kind of the study setup is hard to do in the humans. You cannot invite the people and say, oh, we will feed you high-calorie diet and see whether our treatment stops that or not. So in the real scenario, in the translational point, it's not feasible. So to make it more translational feasible, what we did is we took the obese mouse, which are already obese, because that's easy for asking the people who are already obese and see if they want to try something which might help them to reduce the weight. So when we took this mouse, what we did is we switched these half of these mouse on the low fat diet and half of them still keep on the high fat diet, and feed them this VSL3, which we found in the earlier study is effective to suppress the body weight gain. And you can see here when we put the VSL3, the mouse on the low fat diet lost more weight. However, on the high fat diet, also maintain the body weight as similar as when they are on the normal chow. So means they don't gain the weight even though they are on high fat diet when we started the VSL3. So that's the kind of shows the protective as well as the therapeutic effect. And that was kind of similar pattern you can see here in the fat mass was also significantly reduced in the VSL3 treated mouse, suggesting that the overall adiposity and obesity phenotype was improved. Question comes is, how does this really probiotic is doing this? So we were monitoring the high fat diet, food intake. And you can see here somehow the VSL3 fed mouse are eating less. So if you look on the calorie intake, they are eating less in the both of the studies. One of the things I would like to highlight here is they are eating less, but they are not eating less than the normal chow. So they are eating the equal calories what the normal chow are eating, even they are offered for the high fat diet. So somehow they are not like kind of starving, but they are sensing the energy is better, what they need. And that's all they are eating, whatever is needed. And the mouse models are much more better system than the human to doing this, especially the food intake studies, because we have a schedule. We will eat breakfast, we will eat lunch, we will eat dinner, right? But mouse only eat when they are hungry. So it's a kind of the hunger-derived phenotype here, and the food is freely available for them. They are eating only how much that is needed. So kind of it's very interesting to see that is they are not starving, but they are eating less and sensing the calories very efficiently to just maintain their normal body weight. Now, when we talk about how does this the food intake works and how does this VSL3 can impact this whole food intake behavior. Now, when we look on the food intake behavior, normally the adipose tissue derived leptin is the one of the major adipokines which plays the role in the food intake behavior. So when we eat something, our adipose tissue sense the food energies and then release the leptin, and then the leptin goes into the brain and suppress the food intake. So leptin is very efficient on regulating the food intake behavior. So when we measure the leptin levels into the VSL3 treated mouse, and you can see here the leptin levels were increased as compared to normal chow. That suggests that VSL3 might be promoting the leptin, and that might be reason why these mice are suppressing the food intake. It's just kind of another overview of how does the leptin work. So when the leptin gets released from the adipose tissue, goes into the brain has the receptor for leptin. So brain cells has the receptor called the leptin receptors, and they follow a signaling pathway through the STAT3 phosphorylation. And ultimately, when the leptin is there, that decreases the expression of the AGRP and NPY proteins in the neurons, and that reduces the hunger. So these proteins, when these proteins are high, these proteins increases the hunger, but leptin suppresses them. And this POMC is the another opposite function protein. Is it basically increases the it is high when it is high when we are satiated or we are full. So that's how, like, nobody, as I said in the initially, is when we are hungry, nobody tells us you are hungry and then go and get the food. And when we start eating the food, nobody tells, oh, you are full and you should stop. We stop automatically. So these are the mechanisms in the brain is happening, and our brain tells to our mouth or gut, okay, stop eating or start looking for food. So when we look on this signaling pathway, especially the STAT3 in response to leptin, and you can see VSL3 has higher STAT3 phosphorylated status in the hypothalamus, which is major center to regulate the food intake, as well as the looking this AGRP, NPY, and you can see the AGRP and NPY is decreased, and POMC levels are increased into the VSL3 treated mouse. So this is suggesting that the VSL3 is able to change the brain axis to regulate the food intake. Now the question comes is it really the VSL3 is working through the leptin pathway? So what about if we take the leptin deficient mouse and see that whether VSL3 effect is gone away from them? What we have seen is actually if we feed the VSL3 to leptin deficient mouse, which is called the LAP-OB mouse, the leptin is, VSL3 is still able to decrease the body weight or obesity in the leptin deficient mouse, suggesting that leptin is not the only mechanism where the VSL3 is working. So then what are the other mechanisms actually are involved? So the question comes the next. And now when we talk about this, another dynamic of when we eat the food, what happens is when our stomach is empty, then the gut hormones called the ghrelin is mostly high. And then the other hormones like the cholecystokinin, GLP-1, PYY, most of them, they are low. But when we start eating and when our stomach is full, the ghrelin levels goes down, the appetite, that brings the appetite down. And then the CCK, GLP-1, PYY goes up. And that's how our brain get the satiated signal. So we wanted to look on this, what is happening on the gut hormone on the VSL3 treatment. we run the whole panel of the gut hormones, and what we found is the GLP-1 levels were significantly high in all these three models what we have tested, whether it's a preventive study where the VSL3 treated mouse has the more GLP-1, or whether it's a therapeutic study again, or whether it's a LAP-OBOB mouse. These all has the higher levels of the GLP-1, and GLP-1 is known to reduce obesity and diabetes. GLP-1 agonists are already available into the market for treatment of type 2 diabetes. They also known to reduce the body weight. So that makes sense. The GLP-1 is high, that will take care for the obesity and diabetes, and GLP-1 is known to also decrease the food intake. So that basically ties up here. How does the VSL3 can reduce the food intake? Now the question comes is, how does these bacteria living in the gut or derived from these probiotics can change the production of these gut hormones produced by our cells? Because the fact scenario is bacteria, although they live in our gut, but they normally do not touch our cells. So one of the reasons is because they are always separated by a glassy layer called the mucus layer. So they can be colonized on the mucus layer, however, they cannot touch our cells. So then how they communicate? They mostly communicate by producing certain type of metabolites. So we started looking on the metabolomic studies, and what we found is when we look on the, especially the short chain fatty acid profile, which is the major metabolites produced by the microbiome. And you can see here, the VSL3 significantly increased a metabolite called the butyrate in all of these three models. That shows that somehow the probiotic of VSL3 is promoting the production of this butyrate in the gut. And that production of butyrate might be the one which might be responsible for interacting our cells and producing more GLP-1. So we also verified that in the cell culture system, when we put the butyrate in the GLP-1 producing cells, it spikes up the GLP-1 production. So that proves that is the microbiome produced butyrate can increase the GLP-1 production, and then GLP-1 can be responsible for this lean phenotype behavior or less food intake here. So we draw the model is basically on high fat diet. It's known that the high fat diet will change the microbiome, but when high fat diet is combined with the VSL3, it maintains the healthy microbiome by producing the more short chain fatty acids, especially the butyrate. This butyrate interact with our L cells, which produce the GLP-1, and then because the higher GLP-1, it maintains the lean phenotype behavior. So now the question comes is, okay, then why we have to go the alternative route, get the probiotics and then produce the butyrate? Why not to treat the obesity directly with the butyrate or not? We tried the butyrate itself, and it does work. It does reduce the obesity and diabetes, and how it basically comes on the same mechanism what we have discovered earlier with the probiotics is, but with the more mechanistic inside, what we found is when we feed the butyrate, creates a situation called the gut tissue remodeling, where it increases the cell proliferation in the gut cells. That leads to include the increased cells which produce the GLP-1, and these cells are L cells, as well as it also promotes certain types of neurons in the gut called the nitric oxide synthase neurons. These are the inhibitory neurons, and these neurons, what the role of the neurons is it basically plays the role to food movement. So for example, when we eat the food, right, if our food moves more quickly in the gut, our stomach become empty very quickly, and we want to eat the next meal. But think about if the food moves slowly, we mostly will feel less hungry, and that's how we can have the more gaps in between the meals that will result ultimately the less food intake. That's what exactly is happening here is, and the butyrate reduces the gut transit for the food, and that's how the food remains longer, and that is ultimately sends the signals to the brain, because stomach says, oh, I'm full, gut says I'm full, no need for food, and that creates the satiety signals and reduces the hunger signals, and that ultimately result to decrease the food intake and the lean phenotype. So basically that's the mechanism of what we find further for the butyrate. However, looking the more feasibility to go the clinical studies, especially for the diabetes and the obese people. So when I started looking on the kind of doing a clinical study and talk to the clinicians, although the butyrate formulations are available into the market, so you can take the butyrate and these tablets and can give to the people. But looking on the especially the obesity and diabetic patients, doctors do not want to put them on the butyrate formulations, especially the currently available. The reason is because most of these butyrate formulations are present in the salt form. Either they are sodium butyrate, potassium butyrate, magnesium butyrate, calcium butyrate, you name any kind of the combination. And the dose what we give is around 4 to 5 grams per day. That's kind of the dose comes into the mouse to play the role in this. And if we put that much, that means we are putting this much salt, extra salt every day to the patients also. And then physicians don't want to do that. So basically then that options is especially for the diabetic and obese population is kind of blocked. So we went back to the probiotic approach. Now because we wanted to go to the VSL3, but then VSL3 is a commercial formula. They don't want to disclose what kind of the bacteria they have, what strain it is, and what it does. Really, all those questions remains there. Also, we also don't know from where those bacteria are coming from, where they are isolated, no origin, nothing. So what we tried to develop is our own consortia of the microbe, and especially selecting them for the butyrate producing. So what we did is we designed a strategy to screen these bacteria from the infant diapers to make sure they are human origin, first of all, because that's required in the probiotics. And then selected them based on they should be glucose utilizers, because diabetes people and obese people will have a lot of glucose flowing around in the gut also, because we measure the glucose in the blood in diabetes, but its reality is glucose is high in all the possible fluids in the body, including the digestive fluids and all. So the glucose will be high into the gut also. Most of these bacteria what we select also are the lactate utilizers. So they can utilize the lactate, because mostly glucose utilizers will produce the lactate. And ultimate, our goal is to put this as a community and then have the higher production of the butyrate. And we started calling that as a probiotic mini microbiome approach. And these are my excellent postdoc fellows are working on these projects. What we found is the first question was, as I said, why we are putting these complex versus the simple probiotic composition, because we wanted to see that is whether the only lactobacilli combinations works better, or enterococci what we selected for, or if we combine them together, that works better to modulate the microbiome. And you can see here, we have done the 5-dose studies. Like for example, we have given from Monday to Friday, the 5 days, this bacterial dose, and see if we treat these mouse with these probiotics, how long this effect sustains on the microbiome. And you can see here when we feed these mouse with so definitely in control, there is no change in the microbiome during the 1 month. But when we feed them with the lactobacilli, you can see that at the 0 time point versus the 2 week, 3 week, like after 5 days as well as 5 weeks, there are changes are happening on lactobacilli. These are the 5 lactobacilli, and these are the 5 enterococci. You do see the changes happens there, but most of the changes comes back closer to the normal. However, when we make the cocktail of these 10 bacteria or the 5 lactobacilli and 5 enterococci, they change the microbiome much more prominently as compared to their own single changes. That shows that the combination can work better than the single form of the probiotics are there. And you can see by signatures also, you can see the most of the changes are happening in the combination as compared to their single formation. When we look on the butyrate and this metabolite production here. You can see here, mostly the butyrate levels are not much changed into the single formulation, but you can see when we put in the cocktail form, the butyrate levels goes up, but after some time they comes back down. So this tells the two story. One thing is the cocktail of the probiotics can work better. The second thing is the changes what the probiotic cocktail makes, they are not for forever. So if you take the probiotics for a few days, it's not like your microbiome is changed for forever or metabolite production also changed for forever. No, this will come back after some time because there is a washout effect will come out. So that means it has to be taken more frequently. We also did the one dose study. So like, see if somebody takes the probiotics just only one time, how long that effect remains. We mostly find out is if we give the one dose, the effect remains for 3 days. So that means that every third day somebody has to take the probiotics if they choose to take the probiotics to maintain the effect on microbiome, maintain the effect on the butyrate levels. What we also found is it's not just only in the mouse. This probiotics also change the microbiome into the humans also. So we have formulated a in vitro system where we can culture the human microbiome here, and you can see here the human microbiome does change over the time in the cube. But if you put these probiotics does change maintain the microbiome diversity much more significantly different than the controlled deterioration, and it does also increases the production of short chain fatty acids like butyrate and others. We have published this story back in 2018, and by concluding that single and the 5 days dosing of the this human origin probiotics cocktail modulate the microbiome in human and mouse both. Effects are not for forever, or they are transient and they are reversible. So that's a good and bad for depends like how you take it. Combination created has the better effect. As I said, the cocktail is much more effective to change the microbiome and the butyrate production than the only single species cocktails. Probiotics, as I said, increase the butyrate. He also asked another question is, are there other ways to modulate the microbiome and increase the butyrate? The other ways is the another strategy called the prebiotics. The prebiotics are mostly the kind of the food for microbiota. Mostly the good bacteria utilizes these prebiotics. And prebiotics are kind of the complex carbohydrates which are not digested by our digestive enzymes, but they can be fermented by the bacteria living in our gut. So they are specifically are available for our microbiome rather than our own digestive system. So to isolate certain types of prebiotics, what we adapted is one of my PhD student from Iran. She isolated the prebiotics from 5 different food ingredients like acorn, quinoa, sunflower, pumpkin, and sago. And these are the final products you can see here. We choose based on the yield because we want to do the mouse study, so we want to get the higher amount. Also, we put the criteria of how pure they will be. They should have least contamination of the proteins, alkaloids, flavonoids, whatever is coming from their source should be the least. And based on that, we selected the acorn and sago polysaccharides and then did the mouse studies. So what we found is, you can see here, and we also compare with another prebiotic which is very commonly used in most of the scientific studies called the inulin, and see how does our new prebiotics compares with that effect. So you can see on the microbiome, all these 3 prebiotics change the microbiome, but change very distinctly. Looking on the effect, like specifically on the phyla level, you can see here the bacteroidetes levels were significantly high into the sago, also high into the acorn, much more prominent than the inulin. And bacteroidetes are kind of a good bacteria because in obesity and diabetes, these bacteroidetes levels goes down. and that's and our mouse are obese and diabetic. So this feeding of this prebiotic is recovering the microbiome which were lost during the obesity and diabetes. This is a good thing is happening here. And these are kind of the overall changes are happening in the microbiome. And you can see here, the inulin is causing the blue signature. The sago is causing this more purple kind of, an accord is causing the red. Accord shows the least kind of effect on the overall signature, but you can see sago and inulin is very effective to change the microbiome. Now, looking on the pH levels in the gut, because if the fermentation is happening, is mostly that will reduce the pH into the gut. This is kind of a good thing happens into the gut, and all these three prebiotics are able to reduce the pH, suggesting that these all three prebiotics are getting fermented by the microbiome. And then again, if you look on the metabolite products, and you can see here propionate, butyrate, and acetate, they all get increased by these selected prebiotics, suggesting that the fermentation is happening for these prebiotics, as well as they are producing the butyrate and other sorts and fatty acids. Now, looking on the effect on the diabetes and metabolic phenotype, the glucose tolerance is the gold standard procedure to measure the glucose tolerance. Here you can see here these prebiotics actually also improve the glucose tolerance, as well as especially the acorn polysaccharide improve the insulin tolerance, which suggests the insulin sensitivity. Now, by looking on the histology of the adipose tissue, you can see here most of these prebiotics fed a mouse has the smaller adipocytes, which is a good phenotype. And also you can see here the mouse fed with all these three prebiotics completely stop the fat accumulation into the liver, which is called the non-alcoholic fatty acid liver disease, which is common in most of the diabetic and obese people, is stopped there. It has the overall health beneficial phenotype there. Now, there is another condition also comes with the obesity and diabetes is called the leakiness, because one of the condition is in diabetes and obesity is increased low-grade inflammation, but nobody knows where this inflammation is coming from, why this obese and the diabetic body is inflamed. One of the condition is mostly obese and diabetic people has the higher gut leakiness. So gut leakiness allows to non-specific leaking of the microbial antigens of the gut, as well as the antigens coming from the food that can stimulate the inflammatory signals. And this process is constantly happening in the diabetic and obese gut. So that maintains the constantly high levels of the leaky gut condition, as well as the more low-grade chronic inflammation in the long term, which basically messes up more with the obesity and diabetes pathology. So, and you can see here in the control mice fed with the high-fat diet have very high leaky gut, and these prebiotics could suppress the leaky gut very significantly by increasing the tight junction proteins, because these tight junction proteins are the one which actually holds the intestinal cells much more tighter. So that's how that reduces the leakiness. And this protein is called, one is called zonulin, another one is called the occludin, and all the expression of all these two genes, both of these two genes were significantly increased upon the prebiotics. We also look on how this, because as I said, we started with the gut-brain axis. We looked the effect on the hypothalamic gene expression of the hunger inducing genes versus the hunger reducing gene. You can see here that there are marginal decrease in the AGRP and NPY neurons and increase into the POMC, but not much significant. So looks like there is a change happening in the gut-brain axis, but not as prominent as the butyrate and the VSL3 can do by these prebiotics. So based on these results, what we have concluded is if we take these prebiotics, either inulin, acorn, or sago, they are fermentable by the microbiome. They produce the short chain fatty acid like the butyrate and propionate. And these butyrate and propionate decreases the gut permeability, and by increasing the tight junction proteins, that result to decrease the inflammation, as well as also modulate the by increasing the POMC into the brain, and ultimately they increases the insulin sensitivity and decreases type 2 diabetes and obesity into the mouse models. Now, again, like when we look on the more translational view of translating these findings to the clinical settings, it always comes as, okay, now you have these prebiotics, now you have those probiotics. How are you going to feed these to the people? To make it more feasible, what we went ahead is we developed a symbiotic yogurt where we combine the probiotics and prebiotics together. And this yogurt is ready, and now we are doing some studies into the mouse, and definitely we are starting the clinical studies to modulate the microbiome and increase the butyrate production. And one of my very hardworking student Rabina has developed this probiotic, this symbiotic yogurt there. So we do have this effect, and then another student is working on the mouse studies, and these are the few results are here. When we feed this probiotic, the symbiotic yogurt to the mouse, you can see here they does maintain the better blood glucose levels. Somehow, or intriguingly, what we found is when we take the control yogurt from the market, it does increase the more blood glucose than even the control, which are non-treated, which was kind of surprising for us. We were expecting like they will have something like either no effect or might be similar effect like what our yogurt has, because we were thinking like that all the yogurt might be good. But somehow it turns out to be very different, that the store yogurt has so now we are thinking and we are talking to these companies what they are adding into, because this yogurt is pretty well matched with what yogurt we made is by in terms of the fat content they have, in terms of the carbohydrates, what they are reporting on the labels, no additives, they are saying it, but we have hard time believing that. The bacteria are definitely different than what we are adding into, and the type of the, I don't know what kind of the milk they are using, but normally they are using the cow milk, what we are using. So it's really not known right now, and we want to do the more kind of the different brands of the yogurt and then compare their effect. But right now, it's very surprising for us is the store yogurt is promoting the diabetes, whereas our yogurt is suppressing the diabetes. Unfortunately, the one what we selected from the store yogurt is actually promoting more than the control, like non-treatment. So it's basically, it's not just a neutral effect. It is actually pushing the more diabetes index on there. So I hope I have given the enough evidence to conclude to draw the few conclusions from these studies. Is the newly developed human origin probiotic mini microbiome modulates the human and mouse gut microbiome and increase the short chain fatty acid, especially the beneficial metabolites like the butyrate. Also, the newly isolated prebiotics from Acorn sago also beneficially modulate microbiome and enhances the production of beneficial short chain fatty acids. Prebiotics decreases the leaky gut and inflammation by improving the intestinal barriers and modulate the gut-brain axis to regulate the food intake and energy metabolism. Symbiotic yogurt is developed in our lab and is ready to go for the clinical testing, especially in the conditions like leaky gut, inflammation, diabetes, and obesity. That's what we are looking. I think so. I'm getting a little late, but I will little bit fastly go on the other topics like the microbiome, butyrate, and leaky gut. As I said, the butyrate promoting bacteria or probiotics or prebiotics are decreasing the leaky gut, but the leaky gut is not only present into the obese and diabetic people, it also presents into the older people. And one of the fact is the older people also has a very different microbiome than the normal people. You can see that here is mouse data as well as the human data combined together. You can see the phyla levels are very different, but on the same time, the older mouse also has the higher gut permeability than the younger mouse, showing that the leaky gut is prominent in the aging. This is the data from the human and mouse together. You can see here, one of the correlates comes is the human as well as the mouse gut has the lower production of butyrate, especially in the older as compared to the younger, suggesting that there is a more leaky gut into the older. There is less butyrate that ties up the butyrate, might be positive or might be beneficial. If you increase the production of butyrate, it might be beneficial to decrease the gut permeability. And this is the direct evidence when we treat the older mouse with the butyrate, it does decrease the leaky gut there. And it does also reduce the inflammation into the gut, as well as we have seen in the brain. It has like whole systemic levels of the inflammation also decreases there. What we also have seen is if we take the probiotics also does do the similar job what the butyrate does, reduce the gut permeability into these older mouse, also reduce the inflammatory marker. One of the phenomena it comes is how does it do is it increases the mucous barriers. So mucous barriers, the part is the goblet cells. You can see these blue cells here. Somehow the probiotics feeding increases the more number of the goblet cells, that makes the more mucus, and that the more mucus makes the gut less permeable. And that's how it reduces the gut permeability and leaky gut and overall inflammation phenotype. So I would conclude based on these results is definitely the older gut have the different microbiome. We call that dysbiotic microbiome or abnormal microbiome, which is associated with higher leaky gut and inflammation. Older gut also produces the less butyrate, and if recovering back the butyrate either by butyrate itself treatment or by probiotic reduces this leaky gut and inflammation and also change the microbiome back. The another topic I would just quickly like to touch is the microbiome signature can be used as a biomarker for different human diseases. One of the example I will present here is we have tested this phenomena into the patients with the mild cognitive impairment, which is the early stage of the Alzheimer, and we would like to kind of predict whether the people has the this MCI condition. And when we look down the microbiome, because microbiome is highly variable to people, and you can see here each dot represents one person. So by looking on the non-MCI versus MCI, there is no overall bird eye view is different. However, you can see there are certain specific bacteria is very different into the people who has the normal cognitive function versus the people who has the MCI condition. And based on that, you can pretty much predict that is who has the MCI versus who has no MCI. So that's how we are developing this phenomenon. When we correlate these microbiome signatures with the Alzheimer's disease markers, which is measured into the cerebrospinal fluid collected from the brain from these people, the A-beta 42, p-tau, and these are the markers of the Alzheimer's disease. And you can see there are certain bacteria which can positively or negatively correlate with these markers can pretty much formulate a biomarker panel there. And we also find out is if we treat the certain conditions or like certain interventions like Mediterranean diet can reverse back these not only the microbiome, it also reverse back the Alzheimer markers also. So the effect of these diets are also depends on the microbiome composition. We also are looking on that some people does improve, some people does not improve. So there are responder people versus there are non-responder people. And one of the phenomena comes is the responder people has different microbiome than the non-responder people. And that is kind of resulting them to respond or not respond for these interventions there. So I hope I have given the enough ammunition or data to show that is to make the overall conclusions here is, first of all, the probiotics are not bad what we have looked on based on the selected probiotics for obesity and diabetes. Even the some probiotics can benefit. How does they benefit is the gut microbiota metabolite got increased by these probiotics and has the several metabolic benefits, like the enhances the GLP-1 production, regulate the food intake by modulating the gut-brain axis, also reduces the aging-related leaky gut and inflammation. Butyrate producing probiotics as well as prebiotics, what we have developed in our lab, has the similar effects what the butyrate itself will have. And then the gut microbiome can be used as a biomarker and to design the precision therapy for the individuals based on the who respond versus non-response. In the Alzheimer's disease, we are also looking how does these microbiome impact what drugs we take, because certain people do not respond for the certain drugs. We are also looking the microbiome prediction for developing the type 1 diabetes, whether we can predict that which kids will develop the diabetes earlier versus later. So I'm sorry, I didn't have time to share that data here. But what I would like to say here is the take-home message is make your gut mother happy and live healthy and longer. I call the gut mother. The reason is because think about our gut is the one organ which feeds our whole body. It gives the nutrients for everyone. So if our mother is not happy or healthy, we should not expect our other body parts to be healthy and happy. So start from there. And I would like to thank my lab members, my mentor, Dr. Rane, and other lab members from his lab, as well as my former lab members and collaborators and funding agencies. And I would be happy to take any questions if you have. Thank you so much.
And anybody wants to ask any question? Please. Anybody wants to ask any question? Quickly ask, please. We are running out of time. Please quickly ask. Or if you want, kindly write in the chat box. Dr. Yadav will give the reply of that queries. Am I right, Yadav? Dr. Yadav?
I'll just have one quick question for Hariom. I know we'll talk a little bit later, but just one quick question. Very nice talk, very informative. So recently, in November, there was a paper in Cell by Zach Knight's group about chemosensory neurons and mechanosensory neurons in the gut, stomach, as well as the intestine. So what they showed very nicely, how just the stretching of the stomach and stretching of the intestine activates the sensory neurons that then go to the vagus, and that's how food intake is regulated. So you're talking about the microbial population in different regions of the gut and in the stomach. So have you looked for sensory neuron markers either in the intestine or the stomach to see whether either adding butyrate or any one of your other molecules that you're testing changes the sensory neurons within the stomach and the intestine?
That's a very excellent question. Yes. What we are doing is right now, we have done a longitudinal study by feeding the high-fat diet from the 1 day to 1 week to 1 month to the several months. And we are doing the RNA-seq on the different tissues, like from the stomach, duodenum, jejunum, and ileum and colon, and also looking the microbiome from these different segments, how does they, and as well as the conducting the metabolomic. Although we do not have the data right now, but we do expect certain genes related to these enteric nervous system will come in that. And I hope definitely I will keep you informed on that part. But we are looking like more global picture rather than looking on the specific neurons. And then based on that, then we will look on the specific gene. So
thank you.
In one way, yeah, we don't have that data yet, but hopefully we will get there.
Thank you.
Anybody wants to ask? Quickly ask, please. I think nobody wants to ask. Very interesting and another topic of great importance, insightful talk, Dr. Yadav. Wonderful talk. We are honored to have you as our guest on this occasion. Your presence on this happy day will be a memory we will always treasure. Dr. Yadav, kindly please stop sharing your slides.
Sure. Thank you so much.
School of Biotechnologies is going to organize BioConference 2020. That is the global meet on 23rd and 24th of August 2020 on digital mode. The aim of this bioconference is to bring together the academicians, researchers, scientists, business giants, and technocrats to provide a forum for the dissemination of original research results, new ideas, and practical development, and to discover advances in the field of biotechnology, management, and education concerning biotechnology, as well as a breadth of other topics. This biotech meeting creates a platform for policymakers, scientists, representatives, and decision makers in biotechnology to present their latest biotech research and learn about all the important developments in biotechnology research. This bioconference mainly comprises of three major categories: research-based, that is research paper presentation, research poster presentation, and third is startup proposals. Under research paper presentation and research proposal, that is research poster presentation, these are further divided into two subcategories: outcome-based research, and second one is hypothesis-based research. Hypothesis-based and outcome-based, these are the two separate categories under these two wide, broad categories, that is paper presentation and poster presentation. And third is the startup proposal. These are the 5 activities of the 3 major categories, which are based on the awards. Those participants who are winning the awards, that are first, second, and third in each category. Those who are interested, kindly encourage your students to participate in such activity. Nowadays and always, there is a need of vitamin P. P stands for positivity. It is very, very essential in this difficult time. There is an end to everything, but we believe that there is never an end, but always a beginning. Thus, to conclude for today, we request Dr. R.K. Garg, Joint Project Director, Center of Excellence in Biotechnology, Madhya Pradesh Council of Science and Technology, Bhopal, deliver the vote of thanks. This is our first venture between School of Biotechnology and Center of Excellence of Madhya Pradesh Council of Science and Technology. He is very humble, very cooperative, and very easygoing person. Over to you, Dr. Garg.
Thank you, ma'am. I deem it is a great honor and privilege to propose vote of thanks on this memorable International Virtual Colloquium on Novel Insight in Molecular Medicine in the Unexpected Pandemic Periods. First and foremost, on behalf of the Madhya Pradesh Council of Science and Technology and Rajiv Gandhi Pradyogiki Vishwavidyalaya Bhopal, I thank our guest speakers, Dr. Sushil K. Rani, Dr. Lalit Singh, and Dr. Hariom Yadav, who despite his busy schedule and found time to grace this occasion and sharing vision and knowledge with us. We are sincerely honored to have all respected speakers. I express and heartfelt thanks to Honorable Professor Sunil Kumar Sir, Vice Chancellor Rajiv Gandhi Pradyogiki Vishwavidyalaya, and Professor Anil Kothari Sir, Director General Madhya Pradesh Council of Science and Technology and Scientific Advisor Government to Madhya Pradesh for their guidance and mentoring to us to make possible this successful event, sir. I owe a special thanks to Professor S.C. Chauvin, sir, Coordinator TEIP, Rajiv Gandhi Pradyogiki Vishwavidyalaya, and Dr. Rajesh Saxena, Senior Principal Scientist, MPCST, for providing technological support and guidance and their liberty to make success this program. And I wish to express my hardcore gratitude to Professor Archana Tiwari Ma'am, Dean Faculty of Applied Sciences and Director, Center for Biotechnology, Rajiv Gandhi Pradyogiki Vishwavidyalaya, Bhopal, who worked hard and ensured that this occasion become a memorable and successful. We sincerely thankful to you, ma'am. I thank all the distinguished team members of MPCST and RGPV who had worked behind this screen of this program. So we all are thankful, our team members of the RGPV as well as MPCST for providing such facility behind of this program. Last but not least, I would like to hearty thanks to all participants who attended this online workshop on the novel insight on molecular medicine, because without good participant, this program was not supposed to be success. Once again, I thank everyone whose contribution has made this workshop successful. Thank you once again.
In a serious note, we would like to request all the participants to please pay homage to our national flag with a standing ovation. One and all, thank you. Now the feedback links are there in the chat box. Please fill up the feedback form and kindly note your name should be clear with your institution name and place, please, because these feedback forms are directly connected to the certificates. Whatever you will fill in the feedback form, that are directly visualizing on the certificate. So please be careful about whenever you are filling up your feedback form, your names along with their institution should be crystal clear. Otherwise, whatever you will just see on your certificates, that whatever you are writing in the feedback form. So please, 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 it is my humble request, kindly in the crystal clear manner, you write your name along with your institution name. Thank you so much once again from the bottom of our hearts of the biotech and MEPCOS families. Thank you, thank you, and thank you.
Thank you.
Thank you. Bye.
Bye. Thank you so much, Dr. Rane.
All right.
Very nice. Bye. Bye.
See you then.
Dr. Hariom, thanks a lot. Wonderful. Dr. Lalit, how are you?
Good.
What
good.
happens to you? What happens to you?
Oh, no, I had some health issues, so I had to undergo some medical surgery.
Which type of health issues?
Yeah, I'm just recovery at home. So with the COVID, I cannot go anywhere as well.
It means I have disturbed you a lot during your recovery period.
But I enjoyed it. Everyone did a good job, and thank you for inviting me as well.
Always. Always, always and always. Only because this is, we all are, as one of the doctoral fellows always says, we all are blessed with the coronavirus. That's why we all are here on the digital mode. One way or other way, we have to just connect with each other. Na? So in this manner, we are connecting. Otherwise, it won't be possible for us to connect in this manner on a same platform, in person.
No, this is really good. This is very interesting with the number of people attending and participating with very active chats. It's really interesting. So now good luck to everybody. Good luck to you guys. Good luck to all the students. You know, be in touch. Thank you again for inviting me.
Always, always welcome. Pleasure is always us. Right?
Bye. See you then.
Bye-bye. Bye-bye. Dr. Mrityunjay, have you pasted the link of feedback forms?
Yes, ma'am.
How many feedback forms have already been filled?
Till now it's 17, ma'am.
Okay. Lines are open up to?
Next 30 minutes, ma'am.
Okay. So shall we wait or shall we leave? We have to wait?
No, ma'am, we can leave.
We can leave?
Yes,
absolutely no problem with the lines?
No, ma'am.
Sure?
Lines are open.
Okay.
Yes,
So
ma'am.
once again, thank you. Yes. Okay. The lines are open till 8 p.m. Kindly fill your feedback form and submit it to you in the crystal clear manner, please. Because these feedback information is directly linked to the certificate generation. Okay? So thank you once. Dr. Lalit, thanks a lot. Your mic is muted. Your mic is muted.
Yeah, thank you. And good night there. And we are going to have breakfast now.
Yeah. Sure. I'm sorry to bother you a lot. Once we will have some queries, we will discuss with you on chat. Okay?
Sure. Okay,
thanks
bye.
Bye-bye. Have a happy and enjoyable day. Stay safe and stay khush also. Stay happy.
Happy, yeah. That's
okay.
Important. Okay,
right.
Bye.
And I am praying my God to the God for your fast recovery, speedily recovery, speed up recovery. Okay?
Sure. Thanks.
Thank you so much. Okay. Dr. Garg, thank you so much.
Thank you. You also, ma'am.
Some participants are there. We will just have one meeting after this, right?
Yes.
Just after this. So Dr. Mrityunjay, shall we leave the meeting? The participants will fill up the feedback form?
Yes, ma'am.
So
sure.
we should leave, no?
We should leave and we are just joining just now for another meeting. Okay?
Yes, ma'am.
Thank you so much.
Thank you.